All right. Welcome, everybody, to Standing for Truth. My name is Donny Budinsky, and it is a privilege to be here with Matt Nailor and Culture Catz for this exciting debate seminar on Adam, Eve, and evidence. This is going to be a comprehensive seminar, and we have a lot to cover. So let's quickly break the ice, get acquainted, and then get right into our first session for tonight. And so, Matt, how are you doing today? Matt is on my team. So we got two teams, team one, team two. We are taking the affirmative, Matt and myself, and Catz is taking the negative for tonight on the topic Adam, Eve, and biblical creation. So, Matt, how are you doing?
Doing good. Hopefully my mic's working. And I'm here at the Tower of Babel, so gonna have some fun. Hopefully everyone's doing good. I'm sure you know who I am on this channel, so no need to introduce. Well, I guess for Cat's channel. Yeah, I run the YouTube channel called Young Earth Creation and teamed up with Donny a long time ago, and we created the Standing for Truth team. So we mostly are known for debating this topic. So.
All right, very good, Matt. I appreciate you doing this all the way from the Tower of Babel. You had to take a time machine to go back there to do it, but that's awesome. I appreciate it. And we're here with our favorite evolutionist, fellow superhero fan of mine, Catz, Culture Catz, formerly known as Conspiracy Catz. Al, how are you doing? I appreciate you doing this.
Oh, mate. Sorry, I just realized I had my microphone turned off. It's always a pleasure to speak to you. You are a genuinely good guy, and I do have a lot of time for you. Matt, we've just met, but I'm sure you're awesome as well. And I'm looking forward to this. And hi to everyone back home on the 810 channel. This should be going out to them as well. I don't know, but anyway, thanks for having us. I really appreciate it.
It's my pleasure. I'm thrilled for this, gentlemen. I think this is going to be really good. I have experience with Catz. He's a good guy. He's informed. He's knowledgeable. And so this is the kind of debate seminar that we are looking for. We've engaged in just countless traditional debates where we've got short openings, short rebuttals, a lot of cross-exam. But we really wanted to dig deep here and give you a good representation of both sides of this debate and this topic. And I do truly believe that's what you're all going to get here tonight. So Culture Catz being our guest, do check out his Culture Catz channel and also his 810 Catz channel where he is simulcasting. And so, okay, we are going to get into our first session as again, we have a lot to cover. And so we want to get the party started. And so what we'll do for each session is have the presenters on screen, and then those not presenting, we will have them backstage. So Catz, we're going to put you backstage for now as Matt and I are in the affirmative, and we have a 50-minute opening statement. So Matt, we're going to be sweating, but this is necessary. So I'll give the first 25 to 30 minutes. Any super chats that come in, Matt, save those at the end of the seminar. We'll spend some time addressing some audience questions. Okay. If no super chats come in, that's okay. Us debaters can ask each other some questions just to have a little bit of back and forth Q&A. Matt, is this coming in good?
Yeah, just make sure you full screen. There we go. Good. And that's us on the side if you want us there.
Yeah, well, all I can see is the screen.
Okay.
So, but you can see me? Okay.
Yes, both of us.
Okay. Here we go, Matt. Feel free to go on mute. And I'll go.
You got the timer?
Yeah, I'm going to start my timer right now. Man, I'm pumped for this. Debate seminar, Adam, Eve, and biblical creation. We are taking on Culture Catz, who I do highly respect. I think this is going to be good. This is going to be one to remember. Resolution. Adam, Eve, and biblical creation are true. Donny Budinsky and Matt Nailor, we take the affirmative. Culture Catz is taking the negative. Here's our website, standfortruthministries.com, where you can find endless content on the topic of origins. I've written several books. Here are the most relevant ones to tonight's topic. This is Special Creation, where we touch on all of these same topics that we'll be discussing here tonight. The Endogenous Retrovirus Handbook. I wrote this book in response to the so-called best evidence for evolution, shared endogenous retroviral-like sequences in the biological world, specifically focusing on humans and chimpanzees, the great apes. In this book, I demonstrate that the best understanding of ERVs is that they are mostly created units of DNA function. As we've discovered, incredible functional roles associated with ERVs. They function in embryological development. They work to determine cell types. They act in cell stress responses in the immune system. As a matter of fact, we wouldn't exist without them. You can get that book and the others on Amazon. Here's an article that I co-authored with Chris Roop. This was reviewed for us in a rigorous review process by Dr. John Sanford, author of Genetic Entropy. In this, we talk about fixation equilibrium. The fact that the observed mutation rate, the neutral mutation rate, even according to prominent evolutionary scientists like Fatuma, the neutral mutation rate equals or approximates the fixation rate. So the neutral mutation rate, as we will see here today, is fast. And those evolutionists that want to appeal to the substitution or fixation rate and say, well, that's a lot slower, we demonstrate here that no, the observed rate, which is fast, approximates the long-term substitution rate. Here's a couple articles that we did. We engaged in a research project looking for in the genome in relation to the human chromosome 2 argument. We searched the genome. Chris Root specifically did the work there using BLAST, and we discovered similar signatures in other chromosomes other than the chromosome 2, where you've got the telomeric repeats in forwards and in reverse, TTA, GGG, CCC, TAA, in mirror form. We found a large signature in the chromosome 9, suggesting that the human chromosome 2 might not be a fusion after all. But we can elaborate on that if necessary. Not sure if that's going to be brought up or not, but if it is, we're ready to discuss it. Overview, real quick overview. In this opening statement, Matt and I are going to discuss several significant topics. These topics include the Y chromosome. This is the piece of DNA that makes a man a man. We will be showing you how science has confirmed the Bible. Science has discovered the first man, and that first man lines up perfectly with biblical origins. All men in the world have inherited Adam's Y chromosome. We've literally discovered Adam and Eve, as a matter of fact, in our genetics. This is why we will also be discussing that little piece of DNA that can be traced back to the mother of us all, Eve. This DNA is the mitochondrial DNA. This opening statement will answer this evening's resolution. Yes, Adam and Eve are real and biblical creation is true. We are going to be discussing the human genome tonight. This is going to be heavy on genetics. What is the genome? The human genome is all your DNA. You can see that you inherit chromosomes in pairs, one from mom, one from dad. Recombination takes place where you get the shuffling of pre-existing DNA variants leading to variation. That's why hypothetically a single couple could have 1 million kids and every kid would be different because we have what's called pre-existing heterozygosity. And so the genome is essentially the complete set of genetic instructions that a biological organism uses to develop and to function. Here you can see the XY, XX. If you're XY, you'd be a male. We're going to be focusing a lot on the Y chromosome. XX, you'd be a female. God is an amazing engineer. A human cell contains 6 feet of DNA. If all the DNA molecules were stretched out end to end. This is all packaged and organized into chromosomes that fit nicely into the tiny cell nucleus. If life were simple, evolution might have a chance. Life is not simple. Life is incredibly complex, which is why the most parsimonious explanation, the best answer to origins, would be special creation, which is our affirmative tonight, specifically the special creation described in the book of Genesis with Adam and Eve. Letters in the genome are incorporated in multiple messages. These are essentially the instructions for building and operating a living organism. A single sequence of DNA can be read in different ways to produce different proteins. And this all evolved over time randomly for no reason at all. We have these overlapping codes. For example, we write books, but we can't write books that can be read both forwards and backwards. DNA can do this. ATCG, every other letter, you could have a third encoded message. That's why the genome is both polyfunctional and polyconstrained, because you get a mutation in a sequence, you're going to mess up several things, not necessarily just one. And that's why we say life could not have evolved through evolutionary processes such as selection, which is a fine-tuning mechanism that keeps a species as strong as it can be. It's helpful, but it's not going to drive fish to fisherman, bacteria-like organism to biologist evolution over time. And mutations, which are primarily deleterious. Most mutations are low impact. Selection can't see them. They build up. Can we design computers in a way that mimics the human genome's sophisticated organization that features what? Overlapping coding regions and regulatory elements? The answer is no. Yet this is what we see in the genome. Modern computer systems, which are highly intricate, lack the inherent complexity and adaptability of biological systems. If we can't design that which is present in the genome, that means the genome itself could not have evolved through naturalistic evolutionary processes. Catz and other evolutionists have quite the task. Why are we, Matt and I tonight, to believe that these incredibly sophisticated systems evolve through naturalistic evolutionary processes over time? Mutually interdependent systems. Why special creation is necessary in light of the data and the facts. I'll go over one. I could go over many, but we've only got time for one. DNA and DNA repair enzymes. It is said that in a single day, we in a single cell could experience up to 1 million DNA lesions or DNA breaks. We have 40 to 65 trillion cells. That's a lot of DNA mistakes in a single day. Thank God we have these amazingly designed DNA repair enzymes that go in and fix a lot of these DNA breaks. Now, yes, unfortunately, some do get passed on in the germ cell line. The mutation rate in autosomal nuclear DNA is roughly 100 new mutations per person per generation, but many of them are fixed through DNA repair enzymes. Now, here's the chicken and egg problem that the evolutionist has, and we'll see how our friend Catz here addresses this tonight. We have the DNA repair enzymes. The information for them are encoded in the DNA. You need both coming into existence at the same time, because if you have DNA without DNA repair enzymes, then DNA is not going to survive or thrive for very long. Will be extinct in a few generations. Now, if you have the DNA repair enzymes, but no DNA, then you're not going to have the information necessary to encode for the DNA repair enzymes. That's why they both had to have come into existence at the same time, which is what we mean by mutually interdependent systems. Okay, let's continue on here. Here's a quote from a famous theistic evolutionist. These are the kinds of arguments that you'll see in an attempt to counter Adam and Eve, the existence of Adam and Eve. So this is Francis Collins, and he said, there is no way you can develop this level of variation between us from one or two ancestors. This argument assumes that all DNA variation is due to mutations. There are two very different positions or explanations for the origin of DNA variation coming from the creationists and evolutionists. As you can see here on the slide, the evolutionists explain the vast majority, if not all, DNA variation as a result of mutations over time. Creationists, we explain the vast majority of DNA variation as being the result of design diversity or created heterozygosity. That is the model that suggests God would have front-loaded Adam and Eve in the original created kinds with pre-existing DNA diversity that can be utilized through genetic mechanisms to produce variation quickly. Now, this model makes testable predictions. One testable prediction has to do with DNA function. And interestingly, ENCODE and various research projects have discovered that at least 80% of the genome has activity. This activity is not spurious. The reason why is because transcription is an energy intensive and resource intensive process. Every time you add a single nucleotide to a growing RNA chain, what you are consuming is energy and you are using ATP. And so if most of this was junk or noise and not legit activity, beneficial activity, the 80%, well, then evolutionary mechanisms over time should have weeded out all of that excess genetic baggage, because it would be wasteful of resources and energy for the cell. And so the best explanation is that this 80% biochemical activity is functional activity, which is why it remains. And we have really good preliminary evidence for genome-wide functionality. We have evidence for functions in just about every single DNA unit or genetic marker that proponents of common descent point to as evidence for evolution, these so-called genetic scars, genetic mistakes, viral DNA, pseudogenes. Here's several papers going over the many functional roles for pseudogenes. They're necessary to sustain healthy life processes in the cell. Endogenous retroviruses, we've identified numerous functional roles for endogenous retroviruses. They're playing an antiviral role in the genome. Were any of these predicted by evolutionary scientists? I'd like to know that. I'd like to have an answer to that. They function in determining cell types and regulating gene expression. ERVs are essential in regulating early stages of human embryonic development. There's one paper that literally says if it were not for ERVs and a very specific ERV, then we as mammals would not exist. I like to exist. Katz likes to exist. Matt likes to exist. Hopefully you in the audience like to exist. And so that's why common design is the best explanation for these incredible DNA units that we find littered in genome. Several more papers going over these functions. And so the design diversity hypothesis has some really good evidence. Okay, the biblical model of human origins. What does the Bible say? Well, really quickly, the Bible clearly teaches that man was created separate and special from any other form of life. Genesis 2:7, Genesis 21 to 22. God created two people, Adam and Eve. And the book of Genesis makes several genetic statements with significant implications. Jesus confirms this. At the beginning, he made them male and female, Mark 10:6, Matthew 19:4. 1 Corinthians 15:45, the first man, Adam. So he's described as being the first man. And Eve in Genesis 3:20, she's the mother of all living. It's pretty clear that the Bible is telling us there's no common ancestry between humans and any other form of life. The question is, do we have scientific evidence supporting that? The answer is yes. We've gone over just some of that so far, but in my last 15 minutes, we're going to go over some more. Overview of the evolutionary and biblical creation models. As any informed person knows, common descent here, this is a sophisticated phylogenetic tree of life, would suggest that all life, including plants, animals, so that would be you as a human related to a banana plant, going back to a single common ancestor. Right here, this is an evolutionary tree showing all life, fish, insects, chimpanzees, humans, algae, all related. The model Matt and I, we are in the affirmative tonight that we are arguing for, defending, is the separate ancestry model. So as you can see here, you've got humans in blue with your extinct human groups like Heidelbergensis, Neanderthals, Floresiensis, Naledi, so on and so forth. We can get into that if necessary. I suspect we'll be focusing mostly on genetics tonight, that humans are separate from any other form of life. I like sticking as close to home as possible. I don't have to show you why humans and strawberries aren't related through common ancestry. I can show you why humans and chimpanzees aren't related through common ancestry. Chimpanzees are said to be our closest cousin. We're supposedly, we go back to a common ancestor roughly 6 to 9 million years ago. If I can show you chimpanzees and humans aren't related, then guess what? strawberries and humans are definitely not related. So we'll stick as close to home as possible tonight. The evolution and creation models, they have a lot of overlap. What I mean by that is there's a lot of agnostic evidence. Okay. There's pieces of evidence that both models can largely account for. If I were to say, Catz, the sky is blue or the earth is round. Okay. As we can see from his background, he's definitely right on that. He would say, well, that doesn't just mean creation's true, because the evolutionary model can also explain why the earth is round and the sky is blue. That's agnostic. That's what's called non-discriminatory evidence. There's a lot of overlap where we have pieces of evidence and observations that both models can account for. Okay. Homology, these shared structures in the biological world. Catz would say that those were inherited by past common ancestors. Okay. You've got variation over time, adaptation. So the structures modify, descent with modification. We as creationists, we would say human engineers design in homologous patterns. Why shouldn't God? What about nested hierarchies? Okay. Humans are a lot more similar genetically, physiologically, morphologically, in anatomy with humans. They got more in common with chimpanzees than they do with monkeys, fish, and banana plants. Okay. Human engineers also design things in nested hierarchical patterns. A sedan has a lot more in common with a hatchback than it does with a boat or a skateboard. And so these are not very helpful. It is interesting, though, in the world of homology and in the world of nested hierarchies in DNA, I wonder why, if common descent is true, why do we have such strong nested hierarchical signatures? Because apparently if we've been accumulating all these mutations for millions of years and most of them are neutral, and it's that neutral variation that selection acts upon to drive evolutionary change, it's interesting that we have such strong signatures, because you think those mutations would disrupt those signatures over time if common descent were true. And so I'd love to see a source or reference that shows evolutionary scientists. I'm not saying it doesn't exist. I'm just interested to see if evolutionary scientists predicted ahead of time homology and nested hierarchies in DNA. Perhaps Catz could provide that. The human germline mutation rate. So selection apparently acts upon genetic variation. The mutation rate is high, but unfortunately for evolutionists, the vast majority of mutations are deleterious. They're damaging, they're disease causing, and selection can't see them, so they build up. Adam and Eve predictions. Low genetic diversity, one Y chromosome ancestor, one mitochondrial DNA ancestor. We're going to be discussing these a lot tonight. Matt also has his roughly 25 minutes to get into these. Low genetic diversity. Okay. You've got two models, the biblical creation model and the evolutionary model. In the Bible, it's clear. God creates two people just roughly 6,000 years ago, Adam and Eve. What that would do is restrict levels of genetic diversity. You wouldn't have a whole lot of genes floating around a population of two. But if evolution were true, and we've been evolving for, let's say, hundreds of thousands of years, going back to the out of Africa scenario, somewhere between 100 and 200,000 years, mutations add something new because they're adding something that was not previously there. It doesn't necessarily mean that that which mutations are adding is beneficial. Could be akin to the dings and rust marks and dents you get on a car, but it is adding diversity. Okay. And so you can even take it back to the Australopithecines. You can see here going back 7 million years ago. That's a lot of time where mutations are adding genetic diversity. And so in the model, the evolutionary model, you'd have erectus apparently living on the globe between 1 and 2 million years. Maybe they exist in a population of 1 million. Okay. Perhaps their generation time is 25 years. That's a lot of genes floating around that population. We should expect higher levels of genetic diversity today. As a matter of fact, I want to ask Catz, where's all the bones? Where's the evidence of this so-called evolutionary history? You got Homo erectus living on the globe between 1 and 2 million years. Let's say they exist in a population of 1 million. 25-year generation time. Where's the billions of dead bodies? Where's the evidence for their existence? The campsites, the civilization, the grave sites, all the dead bodies. You could fit all erectus bones in the back of a car. That tells me that the evolutionary story is not true. Okay. So the separate ancestry model predicts low genetic diversity. The evolutionary model predicts high. levels of genetic diversity. What do we discover? Well, humans are 99.99% the same. Any two human beings, nearly identical in their genetic makeup. And so that makes sense. From the biblical perspective, we'd expect low genetic diversity. That's what we see. We don't see the types of diversity, levels of diversity that if evolution were true, that we should expect to find. Do evolutionary rescue devices work out of Africa population bottleneck? We'll see if Catz appeals to that, and then we will address it. Mitochondrial DNA. Outside of the nucleus, we have the mitochondria. These are the powerhouse of the cell, as people like to hear. You probably know from biology class. These structures carry their own type of DNA, mitochondrial DNA. Now, mitochondrial DNA is inherited from the mother's side. It is uniparentally inherited DNA. And that kind of DNA, its function is it basically controls the conversion of sugars into energy. It creates ATP. And in each cell, you have many little, the number does range, mitochondria that produce energy for us to live and breathe. And that energy sustains life and body function. Notice this. This is fascinating, guys. Genesis 9:19. I definitely want to see Catz address this. These are the 3 sons of Noah, and of them was the whole earth overspread. So the Bible is making some very significant claims that every single human today has originated from 3 founding couples, Shem, Ham, Japheth, and their 3 wives. Those would be the daughters-in-law of Noah. Well, notice this. This is a mitochondrial DNA phylogenetic tree. This represents people. This represents individuals, people groups over time. The lines represent DNA differences or mutations. The tips are individuals. And you'll notice all mitochondrial DNA here can be traced back to 3 main haplogroups. These are not even contested by the evolutionary community. These are the L, M, and N haplogroups. Now, this family tree, going right back to 3 major haplogroups, is incredible because what we just read in the scripture is that all human beings today have descended from 3 reproducing couples. And since mitochondrial DNA is inherited maternally, these 3 major haplogroups right here in the middle, these are these 3 nodes, they could represent Noah's 3 daughters-in-law. And yes, they could live at the same time. They can be live contemporaneously because the lines separating L, M, and N, they're short. That suggests just a few DNA differences. There's only a few DNA differences separating these 3 nodes. The R haplogroup could be a post-Babel dispersal event, but the lines radiating out from them, they're longer, suggesting more mutations, more DNA differences over time, and post-flood history. Guys, the Bible demanded a single woman that every human being descended from. The evolutionary model did not predict this. When we look at worldwide mitochondrial DNA, the family tree collapses rapidly to a single person. This is Eve. The real Adam. The Y chromosome is a master control switch. It influences the expression levels of thousands of genes on all the other chromosomes, from the time a boy is in the womb to puberty and on into adulthood. The Y chromosome is also uniparentally inherited DNA. It is passed down on the father's side. And so, do we have evidence for one Y chromosome ancestor as well? The answer is yes. And we'll see that in a second. I do want to point out that in the mitochondrial DNA, the mutation rates are a lot faster than what's called the phylogenetic approach, what the evolutionists like to focus on. If we dig a little deeper, we'll get into the point that the neutral mutation rate approximates the fixation rate. And I got 5 minutes here, so I'll move quickly and then hand it over to Matt. And so this paper here from 2022 is making it clear that the observed mutation rate is approximately 10-fold higher than phylogenetically derived rates. Notice this from the Parsons paper in 1997. This is roughly, talking about the mutation rate, roughly 20-fold higher than estimates derived from the phylogenetic analyses. Again, 20 times higher. So what we see is low diversity, just a few DNA differences in the mitochondrial DNA, few DNA differences in the Y chromosome, but we have fast mutation rates. In the mitochondrial DNA, the rate is about 1 mutation every 2 to 5 generations. In the Y chromosome, it's roughly 1 to 3 per generation. And so with few DNA differences, low diversity, and fast mutation rates, what this is biblical time. One man, one woman. Notice this phylogenetic tree. This is similar to the one before. This was put together by Dr. Rob Carter, but there's some really interesting things here. So notice this, HVR. This is the most common haplogroup in Europe. You'll notice differing size branches. What this is telling us is that people in the same amount of time, some people are picking up more mutations than their cousins. So that tells us mutation rates are variable. And we have papers, 2022, showing 2 families, germline hypermutation. They're passing on even more mutations. Mutations in DNA repair genes can result in hypermutation. That means the evolutionists, they have a problem there because they are largely going to have to assume a strict molecular clock, a constant mutation rate, in order to take humans and chimps back to a common ancestor, roughly 6 to 9 million years. This is a Y chromosome family tree, same thing. We see people groups, we see individuals, we see one man, we see the reality that in the Y chromosome, we have few DNA differences. If I were to just draw a circle right here in the middle, we do have outliers that we can explain, but right down the middle in here, you're looking at about 200 mutations. Well, with a mutation rate of 1 to 3 per generation, again, that's biblical time. Notice this. This is a paper that just came out. This is incredible. 2025. If you look at the supplementary material, over 12% of the human and chimpanzee genomes represent what's called a gap difference. These are sections that can't even be aligned. So right off the bat, you're looking at roughly 88% difference in whole genome sequencing. And it's interesting because even in your similar sequences between humans and chimps, you have differences in the way that those genes are regulated or expressed. You have differences in alternative splicing, epigenetic differences. There's a whole lot of differences, and it's important. Remember this, and I want Catz to remember this. It's the differences that make all the difference, because creationists, we can explain the similarities. Evolutionists can explain the similarities, but the massive differences, that's where the evolutionists struggle, but the creationist expects. Here's an example. Here's the last point I'll have time to cover. This right here is an evidence that I believe precludes evolution and that Catz is really going to have to address. These are Y chromosomes between humans and the great apes. You'll notice right off the bat, the human Y and the chimp Y. The chimp Y is half the size of the human Y. The chimp has lost the entirety of its heterochromatic arm. This right here, it's all color-coded. The light gray, this is heterochromatin. It's this densely packed kind of DNA. It largely comprises what's called satellite DNA, repetitive sequences. It's not even present in the chimpanzee Y chromosome. And so when you consider overall architecture, size differences, and gene content, the chimp Y and the human Y are less than 30% the same. Actually, we see a break in the nested hierarchy where humans and gorillas in their Y chromosome, they're more similar than either is to the chimpanzee. That's fascinating. And this is 2024. It gets even worse for evolutionists and Catz here tonight. Unfortunately, we've got the most sophisticated DNA sequencing data for the Y chromosomes, the complete sequence and comparative analysis of ape sex chromosomes. You've got human and chimpanzee. You can scroll here and see less than 30% the same. Less than 30% the same. More than 30% of the chimp Y chromosome fails to align to the human Y chromosome. The evolutionary community are forced to say that portions of the Y chromosomes that they've been rearranged. We got gene loss, gene gain, and large-scale chromosomal rearrangements according to them. And so how do the evolutionists explain this data? We'll see here tonight from Conspiracy Catz or Culture Catz, and I'm excited to engage this data further. Matt, brother, feel free to hit unmute, and you've got 20 minutes. Share screen, you're good to go.
Wow. Okay. Well, actually, remember, we were going to promise Catz if you wanted to take a break. You can give me a thumbs up if you want to run away, use the restroom.
Nope,
No, we're good to go.
He's good.
Okay. All righty then. Let me share my screen. All right. Let me see. It's kind of in the way. All right. Does that look good, Donny?
Yeah, that's looking good, Matt.
Okay, I'm going to move some things out of the way and give me a 5-minute warning if you can. If no, that would be helpful.
I went for 30, so we got 50. So you got 20. You got 20 minutes. I'll give you a 5-minute warning.
All right. We're going to start off with the age lifespan, because if these were real people and they're talking about real age limits, then we see in the book of Genesis that they lived for a really long time. And then after the flood, the lifespans dramatically diminished. Now, that's because of something called patriarchal drive, and it had to do with Noah. But what we're going to be focusing on is the lifespans, and we can see them in multiple cultures around the world. So it doesn't just fall into the biblical thing. A lot of people talk about this. Kind of strange that they would invent such a weird thing outside of reality when you want people to believe your religion right out the gate. But what do we know about genetics and the theory of aging? Well, your genes, as well as the mutations in those genes, are responsible for how long you're going to live. So there's a geneticist by the name of Dr. Aubrey de Grey. He's a biomedical gerontologist. He says that humans should live for about 1,000 years. But he's not the only one that said this. There's a book written on it by David Goldhill called Longevity, Why We Are Living Longer Today, and the Discoveries That May Allow Us to Live to Be 1,000. And then we see another molecular biogerontologist talk about how old we could possibly live. And an expert believes we could live for another 1,000 years. We see the same thing. Humans may live to be about 1,000 years. This is by Ray Kurzweil. He's a guy that spends about $2,000 every single month to get there with supplementations until we get the technology to live this 1,000 years. And it's true. You want to live for 1,000 years? Be careful what you wish for. How achievable is this? Well, it's achievable through the field of DNA repair. The mechanism, we are going to be trying to be using something called CRISPR-Cas9. It's a gene editing software where we can erase mutations and go back to the original sequence. And they've done this one time in a gene in mice and in fruit flies, and they call it the fountain of youth. And in mice, just editing one gene allowed them to live 23% longer. And Dr. Valtier said with just a small modification of the genome, you can go from 2 years life expectancy to 100. So is there evidence for Adam and Eve that could live for 1,000 years? Yes. Is there the genetic potential is still inside of us today to reach 1,000 years? So isn't it interesting that the maximum human lifespan estimated by different scientists perfectly land on the lifespan of the limit mentioned in the Bible? What are the chances of the evolutionary secular scientists discovering this and concluding that? So if these experts in their field of genetics are wrong, how and why are they wrong? And what are we doing when we're removing mutations that add lifespan? When we're removing them, what sequences are we mimicking? We're mimicking Eve's consensus sequence. This is the one that's been published in the peer-reviewed journals, and you can read it right here, called Mitochondrial Diversity in the Human Population, published by Dr. Robert Carter. And it's on Creationist website right here, where you can go and you can see the Eve's consensus sequence. And you can see that the gerontologists are using that sequence to mimic. So very interesting. So the goal of this section was to show that there is genetic evidence for long life potential, like the Bible describes, built into us, and it's still there, able to be unlocked through modern day gene editing sequences. I find that fascinating. But what about physical evidence? Again, we are going to go to what were predictions. What did we find? This is one is Jake Herbert by ICR, Institution for Creation Research. He noticed that there's greater longevity with larger adult body sizes and delayed maturation. We're all linked together somehow. So when we go back in time, we want to ask ourselves, is this true? I just ask AI. It says, yeah, slower maturation, reaching puberty is what that means, is often associated with longer lifespans. Well, what's the evidence for that? Easy. We can go to humans. When they reach puberty from 9 to 12, they live 70 to 75 years. When they reach it at 13 to 16, they live 80 to 90 years. And this is through dogs, mice, rodents. You can have AI answer it all. It always is the same thing. Very fascinating. So we want to ask ourselves, how about our supposed ancestors, which we would just say are people that were closer to the flood? Well, they reached maturity at a similar rate or slightly slower. That's with Neanderthals. How about Heidelbergensis? Same thing. It looked like they took longer to reach adulthood. How about bone evidence? Thicker bones live longer. Thinner bones live shorter. Smaller bones live shorter. So we see the same thing. How about bones in ancient archaic human beings? That's right, thicker and more robust bones. Heidelbergensis on this one. How about Homo erectus? Same thing. How about bones in Neanderthal? Same thing. We see the exact same criteria. So when we line them all up, look, modern humans have the smallest and thinnest of all of them. And when we compare them all side by side, we see that clearly our ancestors could have lived longer with an evidence through thicker bones, just like we see in the scientific evidence today. Here's a jaw of an archaic human being right next to a modern day person. So if you want to see the studies that go into it, there's your ICR links for anybody that would be interested. And then we are going to jump into something called genetic boundaries. Now, this is really important. So pay attention here because this is very fascinating. They analyzed barcodes from hundreds of thousands of different species, and they found a telltale sign showing that all animals emerged about the same time. Here's what their charts show. Here's birds, fish, moths. They all form, they all have low ADP, is average pairwise distancing. So they looked in the mitochondria and they looked at the most conserved region, which is cytochrome C. And then they looked in cytochrome C to an even more conserved region called cytochrome C, the CO1 gene. And then so they always look at the first 650 base pairs and go from there. And this is actually a paradox in evolution called the Lewontin's paradox. And it says that the species of vastly different population size exhibit the same similar low levels of genetic diversity. Why? Because not a lot of time has passed. So this is what those DNA barcodes look like. You can look at a species and go, wow, those are very, very different. I wonder if we can use those to determine relations and ancestry. And here's what one of the authors of the paper was talking about. He said, although it's easy to imagine humans passing through a bottleneck 170,000 years ago, it's hard to believe that exactly the same thing happened in all species. Did herrings really pass through an equally recent bottleneck, and anchovies too? I mean, that's a good question. What in the world does that sound familiar to anybody? What? Perhaps a global flood? What else? A global bottleneck could have wiped out all aquatic, all bird life, all animal life, all humans. Come on. They looked in Africa. They said, well, nothing out of the ordinary happened about 200 to 100,000 years ago in Africa. So that nothing there. Then they decided, let's go to the fossil record. Clearly, there's going to be evidence there. Nope. No trace of anything in the geologic record 200,000 years ago either. Young Earth creationists, we had a field day with this. Every organization talked about this study. That's how good it was. And we just loved it because it was the first time ever we have genetic boundaries separating things like the taxonomic boundary paradox, right? Taxonomy places categories and boundaries, but now we have those boundaries in genetics to even prove it. And this upset the authors of that study to the point where they had to add a disclaimer to the top. They say, no, this study's grounded in strong support of Darwinian evolution. I know some people out there say, no one believes Darwinian evolution. Well, they do. Anyway, including the understanding that all life evolved from a common biological origins over billions of years. This work follows mainstream views of human evolution. We do not propose a single Adam or Eve, and we do not propose any catastrophic events, even though they went looking for those catastrophic events. But the problem is that even the news press picked up on this, and they said what we know has debunked much about what we know about evolution of species today. So isn't that funny that they're debunking it in these articles when interviewed? Like right here, another unexpected find in the study was species have clear genetic boundaries and not much in between. The absence of these in-between species is something that perplexed Darwin. Right in between the fossil record, he expected, tons of intermediaries. Can't find them. The conclusion is very surprising, and I fought against it as hard as I could. That's what the authors of this paper said, but yet it strongly supports Darwinian evolution. So they say one thing after the fact because they didn't expect so many people would be like, wow, this is such great evidence for us. There's such great evidence for this independent line of ancestries that we're talking about. And, here it is with wolves. We provide several lines of evidence supporting a single origin for dogs, disfavoring models in which dog lineages arise separately from geographically distinct wolf populations. Exactly what the biblical thing said, right? It would be an unclean animal would only be going on by pairs of two. So we expected a convergence going backwards in time, right? We'd expect to converge on the consensus sequence that we evolved from, chimpanzees and humans. What do we find? The exact opposite. Modern humans have a very similar consensus sequence to the origin of. Some people today have no mutations, and we have a maximum of 10. Then we look up at Neanderthal, they ranged around 8, and Denisovan around 10 to 18, and then Heidelbergensis around 18 to 20. But here's the thing. When you look at those mutations, they're in the same place as the human version. The humans on top, Neanderthals mutated right below it. It's in the same place. And what that means is when you run the consensus sequence through even AI, take consensus sequence from a Neanderthal and a Denisovan, and then revert them, and it matches the human consensus sequence. And that should not be the case if it was on the other side of the bottleneck. There's no way it would converge on ours. It would converge, it branches out from ours. That's the interesting thing. And how do we know that? We can see that when those mutations are removed, it matches our consensus sequence. And it would be the other way around if evolution was true. And that takes us to real quick, does creation model make any predictions about this? Yeah, matter of fact, Neanderthals were predicted way ahead of time by young Earth creationists, by them actually being just regular humans. And that was found recently, which is why humans and Neanderthal are less similar than polar bears and brown bears. And they even admit that humans belong to the same species, could rewrite the history of evolution. And what else do we know about them? Well, they couldn't even have lived for over 10,000 years. How do we know that? Because of their population sizes. They admit that they lived around 10 to 15 people, including children. Some say no more than 20. So let's make a chart based on what that study found. They found that inbreeding with just 10 to 15 people allows a maximum lifespan, maximum 500 years of their existence. If they were between 25 and 50, they can only have lived up to 1,000 years. Well, how in the world could they have lived 400,000 years? It's impossible. And the ironic part is evolution has gotten every prediction it's ever made with Neanderthal completely wrong from its very beginnings. Matter of fact, they were called Homo stupidus in the beginning. So this brings us to our next question. When did this happen? So we found the genetic research going back into that very study, because they just arbitrarily state 100 to 200,000 years. That's why they wanted evidence for it and went to the fossil record, couldn't find any. So the study authors speculated that somehow life nearly got wiped out across the board about 100 to 200,000 years ago and then restarted. So we want to know, when did that happen? Like, what is going on here? And that will bring us into mutation saturation. Over time, mutations build up in the mitochondria. It doesn't matter if they're neutral. It doesn't really matter if they're beneficial. It doesn't matter. They still are building up. They're still occurring. So when did the split happen? They used to say 5 to 7 million years ago, with a maximum of 17. Well, Nathaniel Jensen from AIG decided, let's run those numbers on how many mutations should have occurred over that amount of time. You get an upward limit of 21,450. In reality, we only found 1,483. This is in the mitochondrial DNA, by the way. Not very many. I decided, let's check the CO1 gene. Over time, we've been separated from chimpanzees. About 65 mutations have occurred. So that means that over 7 million years, about 2,450 should have occurred. We're nowhere near that. How about bears? Bears supposedly emerged about 40 million years ago. They should have between 3,000 to 775,000, depending on 4,500 years ago. I did really a big favor by saying maybe they had a common ancestor just about 4.5 million years ago, and then or 40 million years ago. The range still not so good for evolution yet again. You can do the same thing with sheep and goats. Same thing, range completely off. Nathaniel Jensen ran it in flies, worms, water fleas, no matter where you look. How about whales? Same thing. Massive amounts of mutation differences should be there, and they're not. How about in chickens? Same thing, 129 actual differences. Almost 4,000 should exist. You know, evolution says, well, you know, the chicken came before the egg. That's what happens when you ask evolution, right? Science actually says the chicken came before the egg, kind of like the biblical creationists have been saying. Now, the biggest critic of all is named Evograd. He wrote a blog against Nathaniel Jensen. And here's his reply about that very study. He said, well, if that amount of mutations actually did occur, you would only be able to see 12,000, not the estimated 21,000 that he showed on that chart. The 12,000 decisions are still far too many compared to the observed number. I just think it's interesting that Jensen used these silly visual numbers. So don't worry, I removed those silly numbers and just left 12,000 on there. And actually what we found. So even the biggest critic, who's a geneticist, has nothing to say against this evidence. Like, it's literally just ignored entirely. So that's a heck of a challenge for anybody to try to refute. So then we go to, what in the world happens when you type in, how old are humans? You get 300,000 years. But why? Why is that? Why do they give that number? Does that come from the empirical evidence that we get from observed studies? There's only 24 fixed substitutions in the entire human population. No, it does not. It actually comes from calibration of the fossil record. It comes from nothing observed. And I had to double check with AI just to make sure for you guys. Sure enough, evolutionary biologists use the fossil record to calibrate phylogenetic trees. Why? Because Charles Lyell had a secret agenda when he created this a long time ago, and it was to free the sciences of Moses. He was an anti-creationist, and that was his goal. So evolutionary theory estimated about one substitution should arise every 300 to 600 generations. Well, what did we actually find? The very first study to actually come out with Howell, and he found one substitution every 25 generations. Way too fast. But it's okay, because he was looking at a family that might have had a optic neuropathy disease. So maybe just mutations were too fast because they had a disease. No problem. Along comes Thomas Parsons for the forensic FBI. He had access to the blood banks, and he has the ability to look at get blood from all different types of people: African Americans, Caribbeans, Sierra Leone, British Caucasians, Americans, vastly different. He wanted to get a substitution rate, and his results were the most recent common ancestor 6,500 years ago, landing on the biblical timeline. And he used 10 of the top geneticists of his time. And here's his coalescence calculation for anyone that cares on how he got it. And a lot of people say, well, he used the fastest mutation rate. Actually, no, he used the mid-range one. Most of them were actually even faster at one substitution every 17 out of, as you can see here, 73 lineages. So a lot of people go, oh, well, you're just looking at the fastest mutation rates. No. Matter of fact, one of his biggest critics, named Holland, decided to join him the following year and run the math again, and it was basically identical, with one substitution every 30 generations instead of 33. So it actually was faster. So the FBI used his mutation rate today, and they track back and go into known history.
You've got 3.5 minutes, Matt.
Okay, all right, no problem. So what do we know then? I was going to go through more of those studies, but no problem. We have the observed rate way up here with the estimation rates of evolution way down here. They don't match. One's an assumption, one's viewed, one's falsified. All the top geneticists completely agree with this. Here's an evolutionist named Ann Gibbons, and she says that it's now worrying to evolutionists. She is one. She's using the term, so don't get mad at me. It could also complicate the lives of evolutionary scientists who say that the mutation rate should date back to the evolutionary clock, and evolutionists are most concerned about this faster rate. And why? Because using the new clock, she would be a mere 6,000 years old. So, of course, they invoke, well, this is 10 times faster, and there's no evidence of why this should be the case. They've checked for mutational hotspots, selection, random genetic drift. One person decided, I'm going to run just selection to determine maybe selection is what's really slowing it down. He came to the conclusion that, nope, selection is unlikely to be the major cause for this. We just can't figure it out. You need rapid selection. Well, it's unrealistically high for beneficial mutations. You can't get it. Even Kimura back then said it. So if all these experts in the field are wrong, why are they wrong? I mean, they admit that in general, there's a perception and a competition to get an older date, and there's kudos in it. That's why they're trying to push the rate back. It's landing on biblical timelines. Evolution theory is based on mutations. So if mutation rates are fast and they require it to be slow, that changes everything. The predictions are completely out the window. The FBI uses them. They match historical dates. They're used by young Earth creationists to make predictions. They're found in all animals, insects, reptiles, and birds. So you can't say, well, the most recent common ancestor, of course, it's not the first human. Well, now you have to account for why it also lands in all animals and insects and birds that also go back and land on the biblical timeline as well. Good luck trying to do that one. And we also, here's a chicken study. 15 times faster. The substitution rate in avians estimated based on fossil calibrations. What did they find? 15 times faster than they were, just like human beings. 50 generations back, super deep pedigree, 2 different lineages, fast mutation rates. Selection had no effect on them whatsoever. So population growth rates converge on the biblical timeline. Legends of a global flood everywhere. Animals, birds, all have the faster mutation rate. Substitution rates and coalescence point using pedigree studies converge on the biblical timeline. Fast mutation rates and low genetic diversity is a paradox for evolution. It's not for us. It's totally expected. There's no logical explanation why millions of people living on islands uncontacted with human beings 6,000 years ago would have completely died off. And when you ask AI the same thing, it says, yo, you completely need a global bottleneck to reset it. And I just lost my place. Yeah. AI 6,000 years ago, you completely need a bottleneck. Matter of fact,
Matt,
yes,
you've got about 10 seconds, but if you need an extra 5, that's okay. Feel free to finish your final thoughts.
I don't need to. That's good. It's all right.
Okay, Matt, when we all agreed on 50-minute opening sessions, I think we're already realizing that we could have probably doubled that. Both sides, I think. There's so much, and I think Catz is going to prove the same thing. I'm sure he's got a lot to present as well. And so that's 50 minutes. I feel like we just started the 50-minute opening. So to the audience, as we are simulcasting, and so in the side live chat on the StreamYard, I am looking through 2 separate comment sections, saving questions and comments. And so anybody just joining us, this is a debate seminar. And so it's a little different than a traditional debate. It is a seminar, but with 2 sides represented. That's why we have long sessions here. We're now moving into Catz session. But the time limit for our program is about 4 hours. So as long as we're done by 9, we're good. And the sessions themselves are going to take about 3 hours. And so we should have a pretty good amount of time to engage audience questions. We'll bump super chats to the top and also have some time for the guests, myself, Matt, and Catz, to ask each other some questions, perhaps some questions that we feel were not sufficiently answered during the debate seminar itself. Okay, Matt, thank you. To myself, thank you. I'm used to just moderating. And Catz, we are going to throw it over to you. I'll reset the timer. Matt, you and I, we're going backstage. Kat is now the star of the show for the next 50 minutes. I can get your slides up. And you just tell me when you're good to go, Catz, and the floor is yours.
I will do. Before I do that, I just want to say to everybody who's watching on my side, Donny, I've had lots and lots of interactions with him. He really genuinely is a really, really nice guy. I've got a lot of time for him. So I know we were saying before that on the Standing For Truth channel, everyone is going to be slating me and saying what a bad performance, how wrecked I was. And on the 810 Catz channel, it's going to be the opposite way around. But be nice with these guys. Genuinely, I've got a lot of time, a lot of respect for these as people, you know. So they've always been absolutely spot on. So I just wanted to say that before we started.
Catz, I appreciate that. And before I start your timer, I want to say that to my side of the audience as well, because we started off this show saying Catz, our favorite evolutionist. And we mean that. Even though we may disagree on origins, we have a lot in common on other issues. You're easy to get along with. I greatly respect you. And so those in the chat, attack arguments, but don't attack people. Let's respect our guest here tonight. Catz, appreciate that. And I'm going to give you the floor now. You got 50 minutes and okay. I got your slides up. We're going backstage. It's all yours.
Okay. And when it comes to attacking arguments, you're going to see Donny's face on a lot of these slides. And it's not because I'm trying to attack Donny. When you see Donny's face on these slides, it's because I'm quoting what creationists say, and I don't want people thinking I'm making it up. So I'm taking these from videos with, as you can see, a timestamp in the corner. This isn't original in any way, shape, or form. Like I said, I've got a lot of respect for him, but it's just so you know I'm not making up these creationist quotes. I will try and quote my source as often as I can. Anyway, the affirmative claim is biblical creation, Adam and Eve. I'm here to refute that. So I see my job as being 2 things. Job number 1 is to really go at the creationist biblical claim, right? Adam and Eve and creation, to go at that, but also to provide evidence otherwise. I'm going to do both, right? It's not just going to be a hatchet job against creationism. I am going to provide evidence as well. Anyway, here we go. Donny gave this quote in a video, 8 minutes, 50 seconds into this video. Good science always affirms scripture. That worries me. That worries me because the way I see that play out is if it doesn't affirm scripture, it just can't be good science. It has to be dismissible. That's the way I see it play out in reality. Another way, really, of me saying this, or my response would be, by definition, if the supernatural is required to explain your conclusion, it isn't science at all. Case in point here, abiogenesis, and I know that abiogenesis isn't this bit here. All right, but the creationists will believe biblical creation is that Adam came from dust. Okay, fine. If you believe that, you believe that. It is a belief. I have no problem with that. But what I do have a problem with is a large number of creationists not admitting that it's just a belief. There's no amino acids, sugars. There isn't the chemical mixture needed there in a pile of dust to create life. We are expecting supernatural intervention. We are expecting supernatural intervention for a snake to talk. We have no evidence of that. So as long as you accept, okay, that is a belief, that's fine. But when we come to science, now you may not agree with the Nailor experiment. You might think it's not shown what it was meant to show. Obviously, trying to recreate conditions from the ancient earth and show how amino acids can form, etc. And there's been many, many improvements to that. The volcanic spark discharge experiment, deep sea hydrothermal vent experiments, studies of meteorites and interstellar ice chemistry, etc. You cannot argue that the evolution side is taking a scientific approach of trying to find out scientifically what might have happened, as opposed to we're not going to try out at all and figure out what happened. On the right-hand side, we have progress on us getting closer to an answer over decades. And on the left-hand side, we've got 2,000 years of no progress, but worse than that, no effort to explain, just belief. And if you believe that, fine, but accept that it is just a belief. All right, which we've got here. Case in point, Dr. Robert Carter. Now, Dr. Robert Carter has been mentioned. This isn't part of my rebuttal, but he's been mentioned already. He did a paper about, it was mentioned, the patriarchal drive in early post-flood population. He wanted to look at mutation rates in the Y chromosome and see whether it fit the creationist model. But when you actually look at the paper, what he actually did is he created a computer model where he, if I can just resize this properly. In the computer model, he told the model that we're going to be using conditions that will be present after a global flood. We've made our conclusion before we've done anything, before we've done any research. He put that into a computer model, and to make it work so we can model mutation rates in the Y chromosome, the computer model had to spit this out, or he had to spit it in, that fathers could live to 1,000 years old and have children up to, almost 1,000 years old. This is stuff he put into the model so that model could work and say, yes, okay, we can arrive at the mutations and diversity in genes that we've got today if the Earth is 6,000 years old and we take all this stuff you put into account, I can try and find some way to go from A to B. Well, I can do that on ChatGTP. I can put into ChatGTP, imagine the world was created yesterday. What would mutation rates need to be? How old would people have to be before they reproduce? And it's going to spit something out. That isn't science. And we see this a lot. Dr. Robert Carter, I'm sure he's a nice guy, but this isn't science. This is him saying, we're going to start off that the Earth is 6,000 years old, and I'm actually just going to tell a computer to figure the rest out. That isn't science. That is extremely lazy. He put all sorts of stuff in here. We're going to say that each generation can only live 85% as long as the previous generation, for example. Now, I'm skipping through quite a lot because time's going faster than I thought. Another piece of creationist science we're all aware of is Dr. Austin and his Mount St. Helens testing of the rock from the volcanic corruption was only 10 or 11 years old, and it came back at being, hundreds of thousands, if not millions of years old. But of course, no creationist scientist, he didn't tell the laboratory the specimen was only 10 years old. And obviously, potassium argon dating is just completely inappropriate for something so young. First of all, for example, when a rock is formed like that, it takes a significant period of time for any argon trapped inside when it forms to be removed, which is massively going to affect the result. But also, the error bars from the lab were 3 million years. So when this came back at being hundreds of thousands of years old, and all the creationists said, radiometric dating doesn't work because actually it's only 10 years old, what they left out is that it also said it could have been formed 10,000, 20,000, 30,000 years in the future, because that's how these error bars work. It's like using a sundial to try and measure the speed of a 100-meter race. Very, very, very bad science. That is not science at all. But creationists jump on it. And this is where I was going before, because good science always affirms scripture. And those papers that are being, even tonight, we've seen them in the presentation. We've seen Dr. Robert Carter in this presentation tonight. He affirms scripture, so we're going to use him. But it's not science, right? It is very, very, very poor, to say the least. Very poor. So anyway, real science actually starts with evidence and data, and it draws conclusions from it. Case in point here. I'm not going to click on the link. This is going slower than I thought it would, but there's a paper here in Nature, a global reference for human genetic variation. And in it is a lovely little figure here. And what they've actually done is they've taken nuclear DNA, and they've from 26 different locations all over the world, and they've analyzed it for variation, and they've compared it for where the biggest variations are and the smallest variations are within populations. Now, if we look at the out of Africa theory, which creationists cannot accept because that doesn't fit with the model. In the out of Africa theory, we start here with the red arrow, and as the population goes out of Africa, it will start to accumulate mutations. Now, as the population moves on, we might this yellow bit, we might get new mutations here, and we might get new mutations here, and obviously in green here. Now, in the green bit, some of those mutations that they pick up in Australia, obviously will be different to what they picked up here, but they will still share the mutations that they picked up there. Any mutations picked up here will still be shared with this group here. And what that means is this group here left in Africa must, if out of Africa is true, show the greatest variation in nuclear DNA. They must. And when we look at the results of this study, it's quite a recent study. Here we go. Surprise, surprise. Individuals from African ancestry populations harbor the greatest number of variant sites as predicted by the out of Africa human origins. That is science. You get your data, you get your evidence, you look to see if it matches your prediction. What you don't do is create a computer model where you tell it what happened and then just see if it could figure something out to support your confirmation bias. That is not how it works. Okay, let's move on. Anyway, real science as well, like that one we've just seen, when other evidence comes into play, if it supersedes or disagrees with our conclusions, we've got to question it. And if no evidence comes into play and it agrees with it, then it only strengthens it. And we're going to hear a lot about mitochondrial Eve and Y chromosome Adam tonight, an awful lot. But actually, the fact is, whenever you look at studies about Y chromosome Adam and mitochondrial Eve, they all support what we've just seen, that they come from Africa. This guy here, we'll listen to him later on, Mr. Perry, kind of broke the mold not too long ago when his chromosome was analyzed. He was from Africa himself, and it threw back the age of Y chromosome Adam by tens of thousands of years, maybe 340,000 years. Nowhere outside of these kind of models would you get anybody saying 6,000 years. No one without using models like that. You just do not see it. Anyway, moving on. And this is because creationism has self-imposed blind spots, right? We've just had a pandemic where people have been genetically sequencing the virus and using science to create, using genetic science to create treatments for it. We have atomic clocks. We have atomic clocks used for precise navigation systems that require on a really good understanding of radioactive decay. We have all sorts of fantastic technology that creationists will use and trust. However, as soon as the people apply that technology and apply their knowledge to the age of the earth or evolution, suddenly it has to be wrong. It has to be wrong. Yeah, they're brilliant when they do all this stuff over here on the left. But as soon as they start talking about that, they have to be wrong. And they have to be wrong again, because good science always affirms scripture. And when they don't affirm scripture, it's not good. Unless it's what they want to see. So, for example, up here, we can see recent studies by Italian scientists have reignited the debate around the Turin Shroud by carbon dating it to 2,000 years, or using, different aging techniques to get it to be 2,000 years. I saw loads of creationists. Yeah, 2,000 years. Brilliant. Absolutely fantastic. Woohoo. So in this instance, yeah, we'll trust the dating techniques because it confirms scripture. Therefore, that has to be good science. We'll trust that one, but we're not going to trust everything else. Brings me to a story. It's very difficult to debunk anything when you can insert supernatural explanations. For example, this is my mate, Dave. He's an invisible unicorn. Really nice guy, but he's an invisible unicorn. And the other day, I had took my young girl to a party, and I took a bowl of sweets, but I wanted those sweets all for myself. So I put them in the middle of this children's party, and I said to them, please don't eat those sweets. And when I came back, all the sweets were gone. Now, I know these kids are good kids, so they would never eat the sweets. That's the reason I'm using. But I know Dave, my invisible unicorn friend, eats sweets. So it must have been him. In my model, you can't debunk that. You can't try and give me any other reasonable explanation why these sweets might have vanished in front of a load of kids. You might say, well, the kids maybe misbehaved. Yeah, but my model also predicts it, because my invisible unicorn Dave likes eating sweets. My invisible unicorn Dave also farts after eating sweets. And I went to bed the other day, and there's only my wife in there, and it stank. It stank. But my wife said she didn't fart. So it must have been Dave after eating the sweets. It must have been. And you can't debunk that. You can't debunk that at all. Now, what if I take that line of thinking to the geological column and I say, you know what? What if? And there's going to be a lot of what ifs in this presentation. What if the laws of physics change completely so that density doesn't matter anymore whatsoever? Let's take a look at what I mean. I've just taken a sample, just a sample of species here. This is the density of a human femur, 1.8 to 2 grams per centimeter cubed. Now, that is a femur stripped of any skin, soft tissue. The bulk density, when it's got skin, soft tissue around, is less, 1.1 to 1.3. And an entire human body that hasn't been stripped of its legs or skin is 0.085. They span a huge range of densities. So when the flood comes, and so this is true for all animals, right? I didn't want to put all the animals in the list here. That would have taken some time, so I've just put a few. So when the flood comes and it kills all people and an awful lot of animals, obviously, and then their remains, either intact or broken, start to sink down, wouldn't maybe complete bodies go on the upper layers because they are not dense, and, you know, just the stripped bones sink? Wouldn't the bone still with soft tissue attached maybe not sink as much? Wouldn't we find a distribution between layers if it was this rapid sinking? That is obviously what we would see. It's not even a question of would we. We obviously would. Like, totally and utterly obviously, we would see that. But we don't, do we? What we see is a really nice layered structure that isn't layered in terms of density. It's layered in terms of simplicity. Now, I've heard lots and lots of creationist responses to this, but none of them are acceptable, to be honest. We obviously would see a big mixing of different body parts because of the way they sink. You know, and humans here, why do we not see them below, for example, early amphibians? Why do we not see them below them? Why do we see them on top? Obviously, it doesn't make sense. But what if? It's that thing, isn't it? What if my magical unicorn just is like, it doesn't matter. Laws of physics don't matter for this bit. We just forget that bit. It's a huge problem that's never explained. Let's talk about coal. This is Answers in Genesis. Coal, right, is not as dense as a lot of the sandstone that sits above it. Not as dense. Now, the actual model that we look at in mainstream science is that the coal that we get, the sandstone above, or whatever it was, because it was formed from fine particulates, has been compressed over millions of years to become more dense than the coal below. And that's why it's more dense. But with rapid deposition in a flood, we wouldn't be getting that. So why should coal lie beneath more dense layers in the geological column? It shouldn't. But Answers in Genesis, brilliant. I love what they say here. They just say, look, here's our explanation. The earth had loads of vegetation on it. And what happened was that the flood waters, right, they came along and they just washed it all up, and then it sank and formed the coal really quickly. So these leaves and these trees, wood floats. Remember, I've got some densities here of different types of wood compared to humans, for example. Again, wide range. They all, or most of them will float on water, but no, they sank below more dense rock very quickly. But how did they form coal very quickly after defying these laws of physics? Well, Answers in Genesis, again, just make stuff up. I mean, they talk about a team called the Argonne National Laboratory who artificially made coal. They created some conditions, and then at the bottom here, I'll read it to you in case you can't see it. It says, so there is no reason to insist that coal formation requires millions of years. And they say that because here, when it was artificially made, after only 75 days, the original plant and wood material still transformed into coal material. That is what Answers in Genesis say. However, when you actually look at the paper itself, there's an awful lot of stuff Answers in Genesis are leaving out. And that is the fact that the scientists, and there's been many attempts at artificially making coal, not a single one of them, not a single one of these papers says coal can be made naturally in anything other than millions of years, right? What they do say is that in order to make coal artificially in very small amounts, they need catalysts. Clay could be one example. They need very fine control of the temperature and chemistry, and they need pure lignin, not plant matter as it just would be ripped up from the ground. But Answers in Genesis, in order for the explanation of this to work, don't seem to want to include what the team who made the artificial coal was saying. I don't know why, but they didn't. Now, I did say before that we don't see human remains where they shouldn't be, for example, in coal seams. But what creationist arguments will always do is find that one example, a bit like the black swan for flat earth. So find that one example and ignore the fact that it is not a widespread seen thing. It's just a one example, and we've got that one example, and we can just use it to debunk these ideas. And the one example used by creationists for not finding human remains in coal seams, because they are way below human remains, is that they say we did once, we found a couple. I tracked down this news article here. I can't remember now who wrote it, and there have been some human remains found in coal seams, yeah. But actually, what Answers in Genesis also won't tell you is that upon investigation, it's because somebody's fallen down a mine shaft, and then as it's caved in, they become compressed, and the remains have decomposed there. So, anyway, what if? I like these what ifs. Okay, I will get to some pro old Earth stuff in this. I will do, but what if, right? To form the Grand Canyon, rather than have millions of years of erosion, what if the floodwaters were there and then we saw never before seen rates of mountains rising and ocean basins falling to allow water to drain out? And as this water drained out, it, like a dam, burst through this rock in a pattern that resembles modern day erosion, it burst through this rock in this very specific pattern. And what it didn't do is wash all of the dead bodies from the flood with it and put them at the bottom of the ocean. It didn't do that at all. It just rushed out. And then as the ocean basins formed, it lay in that. So again, I can't debunk that. You can't debunk me saying my mate Dave, the invisible unicorn, made that happen. It's not science. Now, I'm not saying that because it's not science, you don't have to believe it. But what I'm saying is, it will be nice to acknowledge that these are beliefs rather than actual science. I'm going to skip on a little bit. Okay. One thing that was talked about here was huge created heterozygosity. I got this from, again, Donny's Adam and Eve, and he mentioned it in the presentation today as well. That Adam and Eve, the way this works is Adam and Eve within their chromosomes, they have all of the alleles, right, that would lead to all the diversity that we see today, or almost all of them, a huge number of alleles. Now, what if that was true? And that's why we had the diversity today. What if they had this unbelievably huge number of alleles? And what if, despite the fact they've had this never heard of before, vast amount of alleles, what if that didn't affect the way the genes were expressed in any way, shape, or form? We've got no mechanism to explain how that would be. We've never seen it before. It's totally at odds at anything that any geneticist would ever tell you. But what if? What if my mate Dave, the invisible unicorn, said it is possible, and what we can do is we can regulate all these genes so Adam and Eve were just expressing a couple as they would as normal people? Yeah, what if? What if that? However, in reality, when we actually look at what happens when you have extra chromosomes and alleles, etc., we have all sorts of problems. Down syndrome, Picoult syndrome, Edwards syndrome. You know, I mean, I'll leave them on screen. We have issues when things like that happen. Serious issues when we just have a little bit of extra coding, unregulated DNA. But are we to believe that Adam and Eve and had all of these extra alleles? Well, whether we believe it or not, it doesn't matter, because about 1700 years later came the flood, and then the population was reduced to just 8 people. So what do we think now? Do we think that even though we're told after the sinning in the Garden of Eden, genetic entropy started to kick in and these alleles started to be lost and degraded, are we to believe that actually 1700 years later, they were still actually all present in the people on the ark? Is that because of reasons? Well, we can believe that because of reasons. But again, I'm using the word belief. And if you want to claim that, but please don't ever claim there's any evidence for it whatsoever, because there isn't. It is literally a belief. Now, something that has been mentioned here, again, tonight, is that when we look at the mutation rates of DNA, they seem quite high, so I'm hearing here, compared to the diversity that we actually see when we look at other animals. And there is a massive reason for that, which I've never heard a creationist address or kind of acknowledge. And the reason for that is that I can't find any species other than human that has less offspring within their lifetime than a human. I can't. Here are some that will have massive genetic diversity. Obviously, every time you have an offspring, obviously some will produce clones if they produce asexually, but a lot produce sexually and have vast amounts of offspring. They reproduce really quickly. How many puppies does a dog have in a litter? A cat? How many offspring can they have over the course of a lifetime? We tend to have what, 1 or 2? Some might have more, some might have none. So of course, every time we have an offspring, that is where this diversity kicks in. So of course, our mutation rate can be high as an individual, but when it comes to passing those on to our offspring, of course, we are going to have less genetic diversity than other species. It's not even a question of why. It's just so obviously, of course, we will. It's not a problem in any way, shape, or form. Of course, we will. Also, we are relatively young as a species, and in the past, we've had a historically low effective population size, which is basically you've got a population, but then those in it that are breeding and passing on the genes has been affected by social issues and social hierarchies. In the past, it'd only be, we think, certain individuals allowed to mate within a group, like we see in the animal kingdom. Populations being spread very, very apart, and obviously bottlenecks due to migration, all to add to what I've just said. Anyway, the Earth isn't flat. We know that. It's not flat at all. For the Earth to be flat, all of this would have to be wrong. I kind of touched on this. All of that, all of physics would have to be wrong for the Earth to be flat. But it doesn't stop the flat earther cherry picking, right? But I saw this, right? That's what the flat earther would say. I saw this, and therefore that's my I'm going to use, and I now know that the Earth must be flat because I can see too far in this one photograph, and I'm going to ignore absolutely everything else. I'm going to take this measurement equipment here, and I'm going to put it down on the ground, and it's flat, so the Earth is flat. And we all laugh at that. We ridicule it. We think it's hilarious. Anyway, the Earth is only 6,000 years old. What would have to be wrong for that to be true? What fields of science? Well, an awful lot of fields of science would have to be wrong. We touched on this before. And what we might do is we might say, oh, I don't maybe have this as my one example, but maybe I've got something like this where I found some DNA in dinosaur bone. So therefore, that absolutely debunks everything else. What I'm not going to do, like flat earthers might do with the black swan, well, flat earthers don't, do they? But I'm not going to look around what the explanation for that might be. I'm going to cling on to this really hard and say that debunks. Yeah, I've heard Matt Powell do this. We found that DNA, that debunks it, done. The DNA shouldn't be there. And we're not going to look at what factors might have led to that and slowed down decay in particular instances. The flat earther might use the spirit level, but like I said before, the creationist, Mr. Austin, might use completely inappropriate dating techniques. So the reason I'm saying this is because what I'd like is for creationists, when they make arguments, to just be aware of how we view certain arguments, and then you can maybe adapt them and word them slightly differently. If you're a creationist in the chat, don't think this is the hatchet job on creation for this first part. I said it would be. I'll provide some pro evidence in a second. But this is how your claims sound to us. So maybe when you're going forward and having future debates, think about, okay, I understand how it sounds for the people. Maybe I can pad it out with, some evidence or explain things differently. I so wanted to read that out, but I haven't got time. Right. And we haven't got sound bites. I like this, though. The creationist soundbite. This has never been observed in reality. You've never observed evolution in reality. That's never been observed. Never been observed. And I love that. I absolutely love it. I've got a couple of other things that's never been observed. And that is death not existing. Again, from this video, I don't want to quote creationists, but I think this was actually from biblical text, not from Donny's mouth himself, but I think he was quoting it from biblical text or something. It wasn't his exact quote. But yeah, death. I've never observed, maybe in a tardigrade, maybe. I've never observed people living this long, ever. And I know that Matt touched on this before, and he was talking about bone density and how long tortoises live. I know this isn't really my rebuttal, but I think you'll find that the reason a tortoise lives longer is because of its physiology and metabolism. Bone density, that wouldn't be the deciding factor. I've never seen people live this long. But you know what? When it comes to science, the Bible says this, so let's believe it. And rather than use geology and radiometric dating to age the Earth, let's use a book that tells us people live unnaturally long lives and use that to date the Earth, which is kind of what we did. I've got another. I've never seen this. I've never seen animals walking, just 2 of them or 7, depending on who we read, out of a population to all group up, all different species, and walk to a single location. That can't happen without divine intervention. So again, not science, divine intervention. Kent Hovind said to me once that the reason kangaroos live in Australia is because when they got off the ark, they were scared of the tigers. He told me that. They weren't scared of the tigers when they were walking towards the ark with them. But anyway, let's get to some pro stuff. And I can go back to some of that stuff later on. Okay. I'll skip all that. Let's go to this. A common claim by creationists is that mutations do not add information to a species, to an organism, or to a species, to a population. Now, that really isn't true. I'll come to this long-term evolution experiment in a minute, but let's talk a little bit about antibiotic resistance in bacteria. And I know what the response is going to be. The response will be, yeah, but we're not changing the kind. Bacteria is still bacteria. But let's have a look at actually how they evolve this antibiotic resistance. Well, they develop pumps to pump the specific antibiotic out of their system. If that isn't new information, a new structure, I don't know what is. They change the structure of their membrane to reduce permeability. They change the structure of the molecules that antibiotics attack, even sometimes going as far as changing the structure of the ribosomes that they contain. If that isn't new information, creating new structures, I don't know what is. Anyway, and we've also, this is well worth checking out, the E. coli long-term evolution experiment. This has been going on for decades. And what you'll find, let me just hide some of these out the way. We've had a sample of bacteria that literally for decades and decades have been just kept and isolated and genetically tested. And what we've found is many, many, many mutations. And actually, if we look here, much of the accumulation was beneficial, not deleterious. Much of the mutation that actually stuck around and carried on was beneficial mutation. Beneficial. You can read that here. E. coli long-term evolution experiment. You can read about it and you can go to it, but much of it was beneficial. Let that sink in. So new information absolutely is being formed if you are forming these new structures, etc. Anyway, but the creationists will say, but there's still bacteria, they're still the same kind. So let's have a look at what a kind is. Well, if you go to Answers in Genesis, they are just species that are created to reproduce within the boundaries of themselves. So basically, they can reproduce with each other. We see that again here. One kind will never turn into another, as evolution requires. In other words, one organism will never become another that can't reproduce with itself. Until we look at ring species like the Greenwich warbler. We look at the population here. This is around the Tibetan plateau. And as the populations go around the left of it, or the north and the south of it, should I say, they started to change, they started to adapt and become different to each other. This population here and this population here can no longer breed. Therefore, by the definition of Answers in Genesis, at least, they are now different kinds, even though they say one kind will never turn into another. But again, the creationists will say, but they're still just birds. And this is a problem. They cannot explain what a kind is. And that is because it's not explained in the Bible, because the Bible isn't a science textbook. What they really are talking about are clades when they talk about kinds like this. And you'll hear Donny and Matt absolutely agree. I hope I'm not misquoting them, but you will hear them absolutely agree in microevolution. They've got no problem with an organism being here and like a wolf, type of wolf, and then splitting into lots of different types of dog. They have no problem with that. The problem they've got, and this is illogical to me, all right? If I just draw this here, all right? This is completely logical to me. They have no problem with an organism here evolving over time or changing over time to become any of these. But they have a huge problem with it going backwards. I don't understand why the cutoff has to be with this one organism here, and it can't just keep going back. I don't understand. That is never explained. But the reason is because we have this text in the Bible about kinds. And that is the only reason a creationist will say, I'm happy with it going this way, but oh my gosh, it can never go that way. Again, obviously not science. Now let's have a look at mitochondria, because this was brought up. And I've got an awful lot of stuff here, but I do want to get to Adam and Eve, because that's really what this is about. I said I was going to go for some Adam and Eve stuff. Right. A lot has been brought up by creationists about Adam and Eve and Adam, Y chromosome Adam. We even saw it actually in, I didn't take this tonight. This was prepared for my presentation. It's not my rebuttal, but I've seen Donny talk about this before, where he's talking about the differences, the huge differences in the Y chromosome between the chimp and the human. And you see, the thing is this, right? It doesn't scream different ancestry. Because if it does, are we saying the more different you are, the more different your ancestors? The flip side of that coin is the more similar you are, the more similar your ancestors. And we are similar in so many other ways. But the real explanation for that, if I can find it, I can't find it, I'll just have to speak. The real difference for that is that the Y chromosome doesn't recombinate. So essentially, when meiosis takes place and you get different chromosomes swapping different parts to kind of like increase variation, you don't get that in the Y chromosome. If there are mutations in a Y chromosome, they are never swapped out because the Y chromosome doesn't do that. So of course, like it's not even a question. Of course, the Y chromosomes are going to differ way more than anything else because that recombination doesn't take place. Also, the amount of sperm that are produced by the Y chromosome, you get billions in a single ejaculate, right? You've got billions of sperm, billions and billions and billions of times has that Y chromosome produced sperm. Obviously, only half of them will be male, but still billions. And of course, when you've got that rapid mutation, the sperm that fertilizes the egg, the chance of it carrying new mutations is massively high, massively high. So this is absolutely what we would expect. Absolutely. Right, I'm going to jump to the end because we've got 10 minutes left and I haven't timed this very well. So let's look at some good science. So some good science would be, I mean, I showed the variation before out of Africa, but let's get to the meat and potatoes of it. We have here a scientific literature review from 2004. What happened was 57 locations across the world were dated for basically historic atmospheric and temperature conditions, right? If we look at the references, the 57 sites, we have them from the 1990s, 1970s, the 2000s, completely and utterly unrelated in every single way, every single shape, every single form, even location. And what they did is they dug down deep to find cores, and they used isotopes, and I'll talk about that if I've got time, if they want to question me on it later on. They used that to determine what the temperature and the atmospheric conditions were like going back in history. Now, here is the literature view, and we can see little graphs, right? It doesn't look too much from where you are, but if I zoom in, what do we see here from these completely unrelated studies? We see an exact copy. Now, what creationists will tell you is that when you use dating techniques, you know, there's errors in there. There's errors, but these studies didn't use the same techniques, right? Some of them used atom trap trace analysis, right? Some of them used oxygen isotope analysis. I'll have to, I'm conscious of time. I'll explain it in more detail later on. Different techniques were used by different people who weren't comparing with each other. And yet, if we're to believe that these dating techniques don't work, why have we got the exact same result here? Why have we got that? We see the same thing here in Vostok and Epica in Antarctica. What happens basically here is there are two different isotopes of oxygen, oxygen 18 and oxygen 16, and as oxygen 18, sorry, and it's deuterium. I'm confusing it with the last one. But anyway, two different isotopes. And as these isotopes make their way down to the South Pole, depending on the temperature, some will precipitate more than others. So what we can, and we know that to be a fact. We know as a fact now, depending on the temperature, you're going to get these some precipitating more than others. So what we can do is we can and we can use this again to as an indicator of historic temperature. And by using other dating techniques from things trapped in the ice, we can get an idea of how old any part of the ice core is. And again, when this is done in different locations, we see we're getting exactly the same thing. I asked Ray Comfort why we get these such similar results. And he was like, I don't know. I like this one. This is my favorite, if I've got time to do it. The Great Pyramids. Quite often I'm told that dating techniques are circular, that they can't be relied upon because you date a tool by the rock, by radiometric dating the rock that it's found in. You can't do that because the radiometric dating is flawed. Therefore, you can't trust the date of the tool and the fossils. And I hear that all the time. It's not quite true. Let's look at the Khufu boat that has been dated to be about 4,600 years old. The Khufu boat in the pyramids of Giza has never been carbon dated. We did use external dating to figure out how old this might be, but not in the way creationists portray it. Number one, we use thermoluminescent dating. I would love later on, if I had the time, to talk just about how good thermoluminescent dating is. It's got nothing at all to do with the radioactive isotopes, nothing. So get that out of your mind whatsoever. The beautiful thing about thermoluminescent dating is it can only date something younger than it is, because the clock resets when something gets exposed to light. And we found that the pottery in the pyramid where the Khufu boat was had been buried for 4,388 years. But that's since the last time it saw light. The last time it saw light. I won't go over how that works. Let's have a look at the others. We saw... here we go. We also use dendrochronology to analyze other wooden structures in the pyramids, and also found out that they were about that 4,500 years old. Dendrochronology has nothing to do with thermoluminescence. They are completely separate techniques, completely, yet they are both these things around about the same time period, which is exactly what you'd expect, given that they were locked away in the same room together. And then also carbon dating, which put it again round about exactly the same time period. And people say, well, carbon dating, you calibrate it against other things. Yeah, let's have a look at that here. The calibration, they said, listen, this might need calibrating because we were 19 radiocarbon years older than, which is roughly, a radiocarbon year is roughly a year, depending on how, like, if you're radiocarbon dating wood and that wood wasn't turned into a table, but it was turned into a table like 100 years before it was turned into the table, then you're dating the tree, not the table. It's but basically about 19 years they estimated, which they may knock on or off. That's pretty damn good. So how do we date this? Right? We use 3 completely unrelated techniques to put it back at a time where there shouldn't be any civilization because the floodwaters were just receding. However, evidence here of centuries and centuries of civilization. Now, my favorite that I'm going to have to go to is isochron dating. I've got 5 minutes left. All right? Isochron dating, creationists hate radiometric dating. They'll always say contamination. If it doesn't fit what they want, they will say it's contamination. I haven't done a good job in the past of explaining this. I'm going to do a better job now. Strontium-87 and strontium-86 are 2 isotopes of strontium. At any one point in time, any one snapshot in time, you will see, and like all over the world, we will have a fixed ratio. Now, that ratio might change over time. Nobody cares. At any one snapshot in time, let's say right now, there's a ratio of 2 to 1. 2 of strontium-87 and 1 to 86. That'll be true all over the world as this snapshot in time. Let's say right now a rock forms. What we can do is we can plot the ratio of strontium. Let's see if I can just blow this up a little bit. We can plot the ratio of the non-daughter isotope, which is this one. This is the one, the 87, that the decay will produce. Right? This one here, 86, is a stable one that isn't produced by decay. We can plot them. And because when the rock forms in this mineral here and in this mineral here, and in this mineral here, when it forms, the ratio is the same. I don't need to know the starting amount. I could have 2 atoms in here of 87 and 1 of 86. I could have 2 million in here and 1 million of 86. Or I could have 4 million of 87 here and 2 million of 86. It's the ratio that's important because that's what we're plotting, and that is on the graph. Now what we do is, and these represent different minerals that will have different amounts. I had this debate with T-Rock. He was insistent, you need to know the starting amounts. I'm making this very, very clear. We do not need to know the starting amounts. We need to accept that the starting ratio is the same, because that is absolutely what we see. The ratio in all those minerals when the rock formed will be the same. Right. Anyway, on the x-axis, we're the parent isotope, which is rubidium-87, which is what is going to decay into strontium-86. Now, over time, as the rubidium-87 decays, right, there will be less of it. And we are plotting this over the strontium-86. There will be more of that. So this number, this ratio gets smaller. We're dividing a smaller number by a bigger number. So all of these are going to shift down this axis over time. But also, because it's producing the daughter isotope, which is 87. Sorry, the 86 isn't being produced. It's the 87, right? Because we're producing that, this number is going to go up. So these are all going to go up as well. But it's going to be in proportion. If there's twice as much rubidium in one of the minerals, then rather than moving, along and up, basically moves diagonally, it's going to move twice as far diagonally in that direction because it started with twice as much rubidium. This one here, if there's 3 times as much rubidium, right, it's going to move 3 times as much. And what we find then is that they will land perfectly on a straight line, if I can do that on PowerPoint. Now, this isn't dating, right? This is checking for a closed system. Because if you can then measure, if you can plot the ratios of these isotopes after time, you know that over time, nothing has leaked in, whether it would be parent or daughter, nothing has leaked out. We don't need to know the starting or the ending amounts, right? As I've explained, we only need to just accept that at any given time, any snapshot in time, the ratios are going to be the same, which is what we see. That is always true, right? And when they form a perfectly straight line, which I'm struggling to do here, then we know it's a closed system, and then we know we can trust the radiometric dating, which is a technique we use to see thousands and thousands of years into the past. This proves a closed system. I had this debate with T-Rock. I didn't explain it well enough at the time. Maybe I haven't. Maybe I rushed. He certainly didn't understand it. He was saying, well, you can make a straight line out of anything. And it was a clunky conversation. I'm more than happy to expand on this in the question and answer session later on if I've rushed this explanation. But until a creationist can explain how this doesn't prove a closed system, obviously, if the line was like this, then it wouldn't be a closed system and you wouldn't trust it. You would know it's contaminated. There's so much more I wanted to say, and I've just blurbed through all of that. Maybe I should have done a bit of a run through. I've got 50 seconds left, so I'm going to do a dance. Dance is done, and I'll stop the screen sharing.
And what a dance, Catz. I can't compete with that.
Thank you.
That was good. Okay, 50 minutes. You've earned yourself a drink there, Catz. That's for sure. And the audience, we appreciate how engaged everybody is and all of the live interaction. We've concluded our opening statements of tonight's debate seminar. Like I said, comprehensive, 50 minutes each. We have many points on the table to rebut and respond to. And even though we have 25 minutes each, I can tell it's going to be fast paced for both sides. So, okay, let's move into our rebuttal. Catz, we'll put you backstage and Matt and I on the screen. 25 minutes. I'll go 12 and a half minutes. We'll split it up. And Matt, this should be good. We got a lot to respond to. So, okay, I'll start right now. Okay, points for rebuttal. I made a ton of notes, and I appreciate just how thorough Catz was in his opening statement. And so we'll take it one at a time. Does good science affirm scripture? Yes, I believe that is an accurate statement. In many ways, the evolutionists could say the same thing. Catz could say, well, good science affirms common descent and an old earth. But the question is, does it? And no, good science affirms biblical origins, as we have seen, and precludes common descent. I think Catz is misunderstanding what we are doing here to demonstrate our affirmative. Okay, we have the data, we have the evidence. We all have the same data, and we are model building. Creationists are model building, evolutionists are model building. We look at the data and we draw conclusions based on it. Okay, I discussed how there's a lot of overlapping evidence where both models can largely account for the data. What we want to focus on is discriminatory data. And I'm glad that Catz provided some evidence that we can deal with that he believes favors his position. Okay, it just turns out that these conclusions line up beautifully with what the Bible says. It did not have to be true that all humans go back to a single mitochondrial DNA ancestor and a single Y chromosome ancestor. If evolution were true, this did not have to be the case. According to the evolutionary model, you have 2 out of Africa events. You have apparently Erectus leaves and independently evolves into other hominins like Heidelbergensis, Neanderthal, Erectus, or Naledi, Floresiensis on the island of Flores, for example. And you'd have people, so historically, random mating is not true. Historically, you'd have people existing in tribes and essentially marrying within those tribes. And so should we find a single Y chromosome lineage and a single mitochondrial DNA lineage? I would say no. We should find many mitochondrial DNA lineages and many Y chromosome lineages. And it turns out, amazingly, that genetic data has connected all human beings back to a single female and single male. These genetic links do not find a match in chimpanzee. Both DNA compartments are incredibly different to the chimpanzee. And again, this data could have come about. We could have went into model building to try and draw conclusions supporting our model, and we could have found genetic data that actually falsifies our model. Okay, so moving on. He said we are forcing a supernatural explanation. That's untrue. We are showing why the data actually demands a supernatural origin for man. Patriarchal drive, yes, Dr. Rob Carter published a paper in the Journal of Creation on that. We cited, at least I cited, his paper in the conventional literature on mitochondrial DNA diversity that shows the Eve consensus sequence, and we're concerned with how long it takes to accumulate the differences that we see, and the average difference is between 25 and 30. He demonstrated that there's low diversity, and with a fast mutation rate, that's biblical time. Doesn't take long at all to accumulate those DNA differences. Patriarchal drive, yes. If the Bible's account of origins is true and our biblical patriarchs lived hundreds of years old, then naturally you're going to be hit. The next generation is going to be hit with more mutations because they are going to accumulate more mutations. Patriarchal drive refers to men, those with the Y chromosome. And so Shem, Ham, and Japheth could have hypothetically been genetic poison to the post-flood population, where you have a whole ton of mutations exploding into the population right away on a phylogenetic tree that would look like a lot of time, a longer branch, even though it's just a short amount of time. He also mentioned the out of Africa theory and why he was moving quick, but assuming he believes the out of Africa theory, the data strongly suggests that. No, I showed evidence for variable mutation rates. I've also got a paper here on my slideshow somewhere showing that certain regions of the genome in Africans mutate faster. And so what Catz doesn't realize is that everything is going to nest within the epicenter of where we all originated or dispersed from. And the epicenter of genetic diversity is right around the Middle East and Africa. But where we actually place that epicenter is based on several assumptions, many of them unknowable assumptions. But all we need is a higher mutation rate in certain African lineages, a selective sweep, where let's say you have a selective sweep across Eurasia that results in older lineages being wiped out, with older lineages in Africa remaining or surviving. Africa could have started with higher population sizes, initial population sizes, where people move out of Babel and into Africa, waves and waves of people into Africa. And Noah's descendants could have brought a lot of genetic diversity into Africa to start with. We also know when it comes to recombination, we have this interesting gene, PRDM9 gene, and that works with recombination. African populations have more of those, and so you'd expect to have more genetic diversity. There's many points there refuting the out of Africa assumptions. Next one, order of fossils. Okay, I would say just overall the pervasive pattern observed in the fossil record is sudden appearance and stasis and extinction. We don't see these large scale changes. Yeah, we could argue about a few interesting transitional forms, but if evolution were true, common descent, we should find millions and millions of transitional forms, not just a few interesting ones like Tiktaalik or Archaeopteryx or the mammal-like reptiles, these interesting mosaics. No, we'd expect to find millions of transitional forms connecting, not these explosions that we find of novel body plans and phyla like in the Cambrian explosion. But I want to see an answer to what I presented in my opening statement, that where's the evidence for the hominin story of evolution? Catz wants to focus on fossils in the ordering of the fossil record. Where's all the Erectus bones? Erectus apparently lives on the planet for 1 million to 2 million years. Let's say their generation time is 25 years. They exist in, let's say, a population of 1 million. Where's the billions and billions of dead bodies? We don't find them. There's no evidence for their grave sites, their campsites, their stone structures. Yeah, we find a couple, which is consistent with an out of Babel model, but we don't find nearly enough that would support the hominin story of evolution. We, as a matter of fact, could fit all the Erectus bones in the trunk of a car. Finding a human bone in a lower layer with a plesiosaur, let's say, would be like finding a needle in a haystack. I wouldn't expect to find it. What does the Bible say? God creates two people, Adam and Eve, and populations of animals in fully working and fully functioning ecosystems. So you'd start off with millions of different species of animals, but 2 humans. 2,000 years comes, you have the flood. The population could have been smaller than most people think, because the Bible does say that there was a lot of violence and wickedness and murder, and that every thought of every man was wicked. Therefore, at the time of the flood, you could have humans in a smaller number in a localized area. That area of the earth could have been subducted with no evidence now, or they were buried and it's like finding a needle in a haystack. But what would we expect to find? Billions of animals. And that's exactly what we find. But this whole argument from the ordering of the fossil record, it's fallacious. Here's an example of what I mean. Coelacanth supposedly went extinct millions and millions of years ago. Okay. And then we find it swimming around in our oceans today, supposedly something that went extinct. And yet we don't find coelacanths buried with humans or whales. Okay, we know they existed at the same time of these ancient creatures. But wait a minute, they did exist this entire time, because we find them living today. And so just because we don't find certain creatures in certain layers does not mean that they didn't exist together, because guess what? Coelacanths existed with ancient creatures that don't exist today, like certain dinosaurs, because we find them buried together. But we don't find them buried with whales, but yet we know that they live together with whales, because we find both whales and coelacanths swimming around today. And so trying to argue that humans evolved later because they're not found with plesiosaurs is simply fallacious. Okay, moving on. Coal, not really my expertise. The debate resolution has to do with Adam and Eve. I am focusing on the genetics of Adam and Eve, but I'll tackle it a bit and then save the rest for Matt. Coal is a fossil fuel formed, apparently, according to the evolutionist, from the remains of ancient plants and animals that lived hundreds of millions of years ago. These remains accumulated in swampy areas that were gradually buried under layers of sediment. And over millions of years, the millions of years assumption, the immense pressure from the overlying layers and the heat from the earth would work to eventually form coal. Well, Catz must forget that in the flood, we would have a great amount of heat, energy, pressure to rapidly form things like coal, oil, natural gas. We'd have plenty of vegetation to rapidly form things like coal. The pre-flood world would have acted as the perfect battery in light of the heat, pressure, and catastrophic effects to create things like coal. And so I don't see that as being a problem. Created heterozygosity, says that there's some problems with created heterozygosity. Well, I went over evidence for DNA functions suggesting that design diversity is true. He'll have to address that and explain how his model can account for all of this DNA function in light of the high deleterious mutation rate that is going to drive species into extinction. Okay, we have about 3 million places in our own genome where our copy differs from mother and father. In about 3 million places, they differ. That's why I said in my opening, my wife and I could hypothetically have 1 million kids, and every single kid would be different because we have pre-existing heterozygosity. And so if we carry 3 million, Adam could have easily carried 10 to 20 million. Today, if I remember correctly, we have 10 million common variants spread out around the globe. And so those could have all been in the ark in Noah's family. They could have been in the population before they spread out. Okay. And Adam, again, could add 10 to 20 million. There's no real contradiction to what we see in terms of allele frequency. Okay. Most of the variation we see in the world is 2 alleles, A or C, G or T. And then after creation, we have thousands of years for mutations to happen to pick up more diversity. He talked about Lenski and beneficial mutations and so-called new information. Well, in every case, in Lenski's experiment, the genes were deleted, broken, downregulated, or eliminated. The majority of so-called beneficial mutations that we observe, they are reductive, functionally compromising to organisms. So it's not necessarily about, well, we want a mutation that adds information. No, we want to see the amount of necessary beneficial mutations that are non-reductive that can counterbalance the damage that's being done due to the massive numbers of low impact, slightly deleterious, nearly neutral mutations that are pouring into the genomes of living organisms today. And so Lenski's experiment does not help. Lenski demonstrated reductive evolution. Apparently, the best way to speed up growth in Lenski's bacteria was to eliminate genes. Necessary genes were removed since those genes slowed down the bacteria, and just more evidence for genetic degeneration. Kinds, we showed plenty of evidence that separates kinds, that separates humans from any other form of life. Y chromosome dissimilarity, I'll have to get to that a little bit later. Dating methods. Matt wrote a book. So I am at 13 and a half minutes. Matt, let's throw it to you, brother. Start right away. You've got right up till 25 minutes. Go ahead. Matt, are you on mute? Matt, I think you're on mute.
Thanks. I was looking at
Okay,
the
so you have
microphone.
So we just hit
My
the
bad.
13 and a half minute mark. So you got 12 minutes. Go ahead.
Okay. Alrighty. First thing I think you should know is that there's 2 different ages for the Earth. One is in a book called the Greek Septuagint. It gives an older date for creation and the flood. So that's why when you're looking at something like an Egyptian pyramid that's around 4,500 years old, that's like, why is that at the same date as the flood? How could there be a population to build it? Actually, they had over 1,000 years to do it. So what we actually see is more evidence for around 5,000 years for pretty much everything down the line, from the dispersion of new harmful mutations arising about 5,000 years ago. We have unbroken chains of genealogies going back to Adam and Eve. Here's a 54-foot parchment at the Lambeth Palace of Library. There's also one at the British Museum for the British Royal List that arose in 1711 or 1171. We have astronomy, goes back about 5,000 years. Medicine, 5,000 years. Irrigation, 5,000 years. Like you said, the trees almost go back about 5,000 years. The oldest desert, about 4,000 years. We have some of the oldest islands that are from sand, go back about 5,000 years. We got the longest living organisms that might not really even die. They're about 4,270, about 5,000 years old. So I like Occam's razor for this because it's the best explanation, is the most simplest, that accounts for all of the data. And just look at everything that matches. We have unbroken chains of genealogies, and that match the genealogical rate, that matches the mutation rate. It goes back about the same time period. Everything just fits so good. Patriarchal drive, it is true, but I would like to say, well, how else would you run a global flood model bottleneck with old people without running the math using that very status? You gotta run a simulation. We don't have old people to put into a bottleneck, even though I'm sure we would want to. They actually used a published study by Kong et al. 2012. It was a secular paper that discovered that the older a person gets, the more mutations they pass on. So all they did is take that and say, okay, let's compare that to Noah if he had 500 mutations and see what would happen if that person had a child. And it created a biological decay curve, exactly what we would expect to find if that was true. It didn't have to be, landed on it pretty good. Another one would be genetic boundaries. I was showing earlier that how would we know what is related to a kind would be a convergence going back in time to a consensus sequence. How do we know that a bonobo and a chimpanzee are related? You go backwards in time to find out if they converge on the similar sequence going back in time. And that is what we see. We can do the same thing with cats. Here is the feline, all of them put together. The pink is the lowest mutation rate found in the species, with the maximum at the very top. We find around 100 or so mutations. That's the maximum in the species. So all cats are within just that window, about 115 and lower. Now, how do we determine that? Because there's some variety, right? There's a nucleotide 58 between lions and tigers, for example, or 58. And then we have 45 between cheetah. So how would we know what's really going on? I'm sorry, panther. If they're related, it really doesn't have to do with how many differences there are. The differences might relate to time, like there's 82 base pair differences between a cat and a lion. But what really matters is there a convergence going back in time to a similar sequence? Because if there is, it had to be on this side of the bottleneck. Because remember, genetic diversity is reset at a bottleneck in the mitochondria. So if everything is converging back to a single line, that's how we know all dogs are related to a single wolf pair. And that's how we can look at cats and say, oh, they fall within the same kind. One thing that did surprise me, though, actually, here's just a picture of a cat. You really can't even tell in the fossil record the difference besides size with cats. They're almost very similar, almost identical. Well, when looking at bears, I was like, okay, well, bears might be all related. It seems like something. I actually found out, no, they're actually 3 different kinds of bears. There's a vegetarian, carnivore, and an omnivore type. So they would actually be separated into 3 separate kinds. So we're getting to the ability now where we can genetically determine what a kind is. And it's great because we've never had an answer for that before. So it's a great thing to do, but it's very time consuming. I literally hate it. I wish I would get some help on it. As far as the fossil record, I actually think that it shows great evidence of a flood, because when you look at the Triassic habitat, you see that was buried first. And then we find a Cretaceous habitat. And then as we see, notice it's being pushed by water towards the perimeter of the continent. And then at the very end, you see it right here. It's being like right at the very, very edge. And then what do we see? Massive depths of sand buried right at the end, like it dumped right in from the continent right into the ocean. And people love the fossil record, but I don't know why. It literally had a single prediction: Darwinian evolution, slow. What did we find? Literally everything else except for that. We found everything: ghost lineages, convergent evolution, stasis, Lazarus taxons, de-evolution, where it goes backwards in time. I think the fossil record is kind of funny. It's interesting to find instead of finding the slow, smooth, progressive change Laurel and Darwin had expected, they found the fossil record rapidly changed, new species appearing out of nowhere, ghost lineages. And what do we see in the fossil record? We always see tracks before we find the animals in layers that can some be millions of years old. That sounds like a flood, doesn't it? Moving up. The only thing that doesn't make sense would be the dates. But first, how do we know that the geographical location can refute this evolutionary paradigm? Easy. We can look at flightless birds. These are ratites, and we know that they've pretty much always been flightless from the fossil record that we find them in. So what would we look at? We would look at their phylogeny that was built off mutations and then where they are living. And to their surprise, the elephant bird are most closely related to kiwis. Completely unexpected and unprecedented. They did not expect this. Matter of fact, the entire phylogenetic chart built off of them doesn't match their landlocked ancestor. Like, for example, you would expect the species that's in Africa, the ostrich, to be more related to what's in Madagascar. But in reality, look at what it actually builds. Completely backwards of what you would expect it to be. Like, everything is completely wrong. So genetics completely refutes that. It's just one of the easiest ways to do it. That doesn't take very long to do. He liked radiometric dating. So let's hop over here real quick. And we are going to go with what is the most important part. He mentioned homogeneity. It's basically is the rock homogeneous inside, because that's what it takes for isochron dating. Well, guess what? There's a study that came out on that very thing, testing 29,000 different isotopes inside there. Now, to run over, this is the margin of error, and right inside of it is lead, and that's 2 different lead dating methods right there. Now, what do we want to find? We want to find concordance. We want to find overlap, and we want them to overlay with one another. That's what you're looking for. What you don't want to see is something outside the dating methods not lining up concordant. So what did the study find? Well, these 2 methods are at the top, are concordant. These 2 methods are concordant. Everything, but as you could tell, everything tested was very almost different from one another. Very interesting, right? Look at this. This is a single rock that was tested with testing different isotopes inside. You can see the score is 0.07. That's not good. The best you can get is 1. And as you can see, there's very little overlap. You can see rubidium and strontium with potassium and argon lined up, but everything else really just didn't do very good. Here's another one, a better score. Here you find uranium and thorium. Oh, look at that. Uranium to lead. 2 separate things, completely off, even though it was tested with the exact same thing. So these are all isochron dates. And even when they are concordant, like this one would be considered a 9.2 score, almost a 1. So it's really good. So even though it scored that high, they were still off by 400 million years. Pretty interesting, right? Here, this one's off by 820 million years. They got a score of 0 using isochron dating. So what did they do? They said, let's compare 3 different types of dating methods to one another and run what it should look like. The score was all over the place. It was so bad that the best it could get out of all methods was 53%. That's a coin toss. That's how bad isochron dating was. And they only tested 3 with one another. Imagine if they tested all of the methods with one another. So this pattern, they also found something very interesting as well. Heavier isotopes tended to yield older ages. Now we're getting somewhere. The systematic discordance is what it was actually called. They found that lead, in this order, lead was lower than rubidium to strontium, which was lower than uranium to lead, or 235, which was lower than 238, which was lower than 232, thorium, which was lower than potassium. Oh, interesting. So is that why they're dating the fossil record on this? Because of atomic weight and not actually because of age? Ooh, I dare say. So some of the results were older than the Earth itself. Others gave negative ages. The majority of rock dating methods all date different from one another. These were called systematic discordances.
2 minutes left.
Okay, there should not be any order to these dates given from different radioactive age isotopes, but there are. So this brings us probably to the most awesome thing. Let me see if I could find it. Where did I put it? Oh, no, I'm wasting time. This sucks. I'm sorry. Hold on. I am so confused. I organized this. Here we go. Okay. Potassium argon dating. What they did is they went to the mid-Atlantic ridge and they went down and they followed lava flow, and they knew that and they tested it, and it was 22 million years old, but the lava was considered to be fairly recent. So they went in the study and they said, look at this. This lava is very young, probably less than 200 years old. So what did they do? They went out another 200 years and they kept going, and guess what they found? The seafloor dated 22 million years old here, 190 million years here, And then right at the very edge, about 380 million, yet it was only 3,408 years old. And then about 4,500 years old on the oldest, at the very top of the largest mountains on Earth, the Himalayas, which dated 500 million years old. Isn't that perfectly in line with the biblical creationist timeline? And it didn't have to be that way. And that's in the published peer-reviewed literature. And since they take radiometric dating and they use it for everything, it's the basis that flows out to calculate with everything else. If it's wrong, that makes everything else that flows from it very wrong. Now, I'm not saying that all of them are wrong. We can look at carbon dating, and it's good. It's good for a certain while. But guess what? This is one of the study where they actually tested it against others. 23 laboratories, or a total of 23 were sent out to 38 laboratories, and they completely failed. They noticed that it was 18% pass. That's atrocious. So we want to ask ourselves, is it really, why do they get dates going further back? It's because they believe in constant uniformitarianism. What did we find? If anything, if there's recent volcanic activity that's going to be devoid of C14, it's going to completely change the date. So we want to ask ourselves, if there was a flood, what would that do? Would it give older dates? Absolutely, it would. So that's the whole question. If uniformitarianism is true, sure, those dates make perfect sense. But if there was a flood that had universal catastrophic things like a volcanism, what would it do? Would it change the dates to make them look older than anything else is? Absolutely. And that's the whole question. Our goal is to be able to answer these things. And we can do that. Okay, sorry.
Okay, Matt, thank you. 25 minutes. That went by quick. So a lot to respond to there. And we're now going to move into the next 25-minute session. To the audience, again, we appreciate all the engagement and interaction from both live chats. I hope people are enjoying this marathon of an event and debate. Again, we should have some time after every single session here. We're digging deep, and I like it, gentlemen. A lot of topics. We should have some time to engage some of our audience questions and perhaps ask each other some questions. Catz, you are on mute. When you unmute, let me know when you're ready. We'll go backstage, and you've got 25 minutes now for a rebuttal.
Well, before you shoot, I think some of the things that you said in your original opening, I kind of had in my opening as well. So what I do have two things that I really, really want to say, but it's not going to take me 25 minutes because I think I've kind of already combined a little bit of rebuttal with what. So what I'll do is I have got a lot of things written down, but if there are specific things you want me to address, if you pop them in the private chat, once I've said these two things, I'll go to those first. If not, I have got loads of stuff that I can use. But if there are specific things that you want me to, I'll jump to them rather than what's on my board.
Okay.
Yeah, same here. Well, I didn't know what to do. That's why I kind of jumped over to radiometric dating for you. I was like, there's too much. There are too many notes.
Yeah, so it's been fun. It's been fun. I should be sharing. Just need it to pop up.
Yes, you're good to go, Catz. You got 25 minutes. Feel free to utilize it however you'd like. And if we got to type anything into the private chat, we'll do so. Floor is yours, Catz.
Thanks. Now, there are two things that I really, really, really wanted to address in my 50-minute segment, but I'd seen them addressed in Donny and Matt's opening. So I thought I'll save the time and do it in the rebuttal. To me, these are really important because it seems like there's quite big things being made about them. So I had this. I haven't just knocked this up now. This was in my original PowerPoint. In this video here, Donny does say, talking about mitochondrial DNA, that the mutation rate is extremely high. Okay. And because the mutation rate is extremely high, if we look at the amount of mutation in mitochondrial DNA at that mutation rate, if we dial it back, then, maybe we can get mitochondrial Eve to be about 6,000 years old. And he's cited this paper here, and he's mentioned it tonight. So both those arguments is mentioned tonight, so it's a fair part of my rebuttal. He cited this paper here by a guy called Parsons, and Parsons did note an exceptionally high mitochondrial mutation rate. And he wasn't 100% sure what was causing it. But he did propose some options. One of the options he proposed was something called mitochondrial bottlenecking during oogenesis, which is basically when the egg forms. Now, one of his hypotheses, I don't know whether it's in this paper or he made it afterwards, is that when the egg is being formed in the female, let's say that during the lifetime of a woman, you have lots and lots of mitochondria in the body. You have this fantastically high mutation rate all over the body. But when it comes to loading these mitochondria into the egg, you might only get one or two of these mutations or none passed into the egg. So although those mutations are happening at a very, very high rate, they're not making it into the egg. And he called that bottlenecking, oogenesis bottlenecking. Anyway, quite timely. I don't know if you can see the date on here, but on the 26th of March, 2025, this was shown to be absolutely the case. So this really does definitively explain why the high mutation rates in the mitochondria do not lead to a 6,000-year-old Earth, if you like. Why we cannot use that mutation rate to dial back 6,000 years to mitochondrial eve, therefore falling in line with everything else. So here we have it. Mitochondrial bottleneck, a process wherein effective mitochondrial content, so that's what's being passed on to the next generation, is drastically reduced. And down here, additionally, random segregation of mitochondrial DNA into daughter cells could lead to the loss of mitochondrial DNA mutations in the progeny. So basically, these mutations are happening at a high rate, but they're not being passed on. And there's a nice little diagram here. I think a primary school person drew this one. It's not something I'd be too proud of, but we're seeing the mutations and then obviously mitosis and cell division, and we're only ending up with, one, if any, of these mutations in the final egg. So this whole argument of this exceptionally high mutation rate, meaning that mitochondrial eve should be 6,000 years old, can now be completely and utterly ticked off as being explained. It was never, and it's fair to say, Parsons never said the Earth was 6,000 years old. He was never a creationist or anything like that. He just observed this high rate and was like, well, how do we explain this? And he didn't jump to any conclusions. If we look at Parsons' paper, that was 1997. And a couple of months ago, it has now been explained. We understand it 100%. So we simply cannot use high mutation rates now as a reason to say that if we use those high mutation rates and track back, then the diversity we've got today would have come in the last 6,000 years. Also adding to the oogenesis bottleneck, we've got to remember that only 3% of mitochondrial DNA is non-coding, right? So 97% is coding. And if you get mutations within those codes that are deleterious, that is going to affect, whether that mitochondria can reproduce, whether that mitochondria is going to be functional, and they're going to be weeded out very, very, very quickly. There's only that kind of like 3% of junk DNA in there where these mutations can kind of effectively go untracked. If anyone's interested, the reason we get a lot of mutations as well is because the mitochondrial DNA is near where the electron, if everyone remembers their A-level biology, the electron transport chain, there's a lot of kind of like oxygen species produced there, and that's why we get a lot of mutations. But explained, not a problem. The next thing that was mentioned in the opening, and again, this was mentioned by Donny, I will try and get to some of Matt's when I do the question, was ERVs. Now, ERVs, it was said tonight, and it's been said lots of times in this video here, in the 2-minute section, that they are mainly created units of DNA function. So let's go over what me and Donny agree on, okay? Which is the model, the evolutionary model. And the evolutionary model is this. We have an organism here, and there is a retrovirus that leaves its DNA in cells that can be passed on to offspring. And I'm going to represent that mutation with a brown dot here. Now, because that's in the offspring and that's passed on, any offspring up this tree here is going to have that mutation, right? So let's just populate this chart here. Anyone's going to have it. I'm just going to go up the, I won't go right to the top because I'm just a bit curious for time. So this mutation, because it occurred here in a region that we passed on in a sperm or an egg or whatever, is now propagating. But on this branch here, maybe this fella now, he gets infected by one of these ERV viruses that leaves its own little genetic marker in a space that's going to be passed on to its progeny. So it's going to be in the sperm or egg. So maybe now this gets a second one, which is going to be passed on to these and to these. And maybe this fella here, he gets one, which is going to be passed on to him and him. And what we can see is we can build some kind of family. Oh, sorry, I've got these as well. We can build some kind of tree. Now, the thing about ERVs is when we discover these and when we analyze these, they fit what we already had as the phylogenetic tree, the evolutionary tree. Right? It fits the fossil records. It fit what we had already determined to be the case with evolution. Now, where was I going with this? Yeah, so it fit that, which would be fantastic evidence for evolution. Now, the rebuttal for this is this argument that ERVs are mainly created units of DNA function. So in other words, these actually aren't due to a virus. These are created by God to have a function, and therefore we would expect them to be where they are. There's two problems with that. One of them is, why do they match the fossil records exactly? Why do they match that as we expect? And also, why, when we actually look at scientific literature, right? Or here they are matching the fossil records. We can see 98.9 to 99% of our ERV insertions are linked with chimps, and we can see it getting less and less as we go to our more distant cousins. And it does track right back through the evolutionary tree in this manner here, this perfectly predictable manner. So how can I refute the fact that perhaps these are mainly created functions of DNA? Well, I'm going to go to the actual literature and actual scientific papers. I'm going to go to this one here from 2018, nomenclature for endogenous retroviral loci. And we can see in this one here, it says that indeed, solo long-term LTR, long-term repeat numbers, are typically orders of magnitude more common than loci containing internal coding regions. Orders of magnitude. What that means is, if we actually look at the ERV base sequence, which we've got here, any bit that actually does anything, right, has kind of been removed. These long-term repeats are just repeating base sequences. That's all they are. They just repeat and repeat and repeat and repeat and repeat. These bits here, because ERVs are being removed, because they just aren't useful, all we're being left with are these bits here. They aren't en masse performing function. Donny's right when he says there are examples of very important adoptions of ERV sequences for placenta formation, for other things. There are absolutely 100% some very, 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 very important adoptions. But to claim in any way, shape, or form that it's the majority to a point where we can say they are mainly created units of DNA function is simply not true. Here we've got orders of magnitude. Those that still remain functional are orders of magnitude less frequent than those that are no longer functional. We've got this one here, another one from 2007. Right, the fate of most of them is to undergo recombinational deletion. So during meiosis, when this recombination takes place, chromosomes swapping data, they're being wiped out. If they were that useful and purposeful, we couldn't stand to have them being wiped out in terms of orders of magnitude more frequently than ones that we know maybe still retain some function. We couldn't afford that to happen. That would be absolutely awful, but it is happening. We've got another paper here from, when's this one? 2015. Nice little quote here. Solo long-term repeats arise by recombination. Let's have a look. Which are much more abundant than the full-length loci. I'm sorry that the text is so small. But basically, it's another paper here again saying, look, yeah, we do have some that retain their function, but it's nowhere near those that have lost their function. Here's another one. Evolution of endogenous retroviruses in the subfamily of Caprinae. And based on our findings, it can be concluded that the majority of ERVs are considered to be fossils, meaning they are remnants. They become integrated into the genomes. All right. However, and then it names some specific examples, which is what Donny says, there are some potentially active ones. So again, if they are created units of function, how can they be being deleted and removed through recombination in a way that they are the ones that retain the function are orders of magnitude less than the ones that have lost it? It just doesn't add up. And here's another one. And you can go through any paper you want to go through. You will never find anywhere outside, I believe, of Donny's book. And Donny, you have to tell me where your resources are. This is a question for you to respond to, not a statement from me. But I believe that you will not find anywhere outside of your book that claims that the majority of them are useful. I believe when you look at proper scientific literature done by geneticists, you will find out, no matter where you look, what paper you look, that it is a minority. So therefore, this claim here that's been made is just not true. They are not mainly anything, although some of them are extremely useful. Yes. Okay. I do have some questions, but I can see in the private chat there's some here for me. I have no idea how long I've got left, actually. So I'm going to...
I can let you know, Catz. So you just hit the 13-minute and 20-second mark.
Okay, so I do have questions to respond to you with, but because there's been a bit of a crossover, if you want me to do specific things, just let me know in the private chat, and I'll do that. Otherwise, I'll just jump to the PowerPoint I've got here.
Did you want me to just verbally,
or you could just ask me verbally? Yeah.
and I'll just ask. I'll go backstage. Feel free to address it. The
Okay.
phylogenetic tree that I presented with the L, M, and N haplogroups, where all the mitochondrial DNA traced back to 3 major groups. I argued that may have been Noah's 3 daughters-in-law.
You did. Now, as far as I remember, and you'll have to tell me if I'm wrong on this. As far as I remember, you've got that from Robert Carter's paper, one of his papers.
No, that one was from Nathaniel Jeanson.
Right, Nathaniel Jeanson. So, without reading the paper, what I'm going to assume, and again, you have to tell me if I'm wrong or not, is that is another paper that has been based on modeling. The similar diagram I saw with Robert Carter's was based on modeling, to which the model was fed the information. That the Earth was 6,000 years old. There was a flood. Was that paper based on modeling, or what was it based on?
Good question. It was an unbiased look because Nathaniel Jeanson and then also Rob Carter with his phylogenetic tree, they just took worldwide mitochondrial DNA data from various databases that you can get that data from, and then visualized it in the form of a phylogenetic tree. So basically that data manifested in the phylogenetic tree. And if you want, I mean, you could look it up. It's not really contested that the L, M, and N haplogroups are your 3 major haplogroups.
No, the data that was fed into Carter's paper, what the data was and it was from a recognizable, valid source. It's what he did with it and how he modeled it I've got the problem with. So the data is fine. And if that is the case, and obviously I've not read it, I'd have to look. But if it is the case that they say, before you get to that mitochondrial Eve, you get to 3 groups, we've got to remember those groups are not individual people. They are not, nobody who's providing this data is suggesting, even mitochondrial Eve is an individual person. Nobody's suggesting that mitochondrial Eve, she obviously is an individual person. Nobody's suggesting she was the only person alive at that time. Nobody suggests that. And she herself would have had ancestors. So I don't have a problem. If that's what it says, that it goes back to before we can hit mitochondrial Eve, it goes to 3 points. I'm fine with that. I don't see an issue in any way with that.
Yeah, well, and I'm not allowed to rebut here because it's your rebuttal, but just to show you the video, I see what you're saying, but it would be unfair for me to respond to what you're saying, Catz. I'd have to save that for my closing. But this is the visual, just for the audience sake. It's these three groups here, L, M, and N, those arrows, that all the DNA. But I understand if that's your rebuttal that you want to go with, that's completely fine. But I just wanted to make sure that you felt comfortable with your response to that.
I'd have to see where the diagram come from, but without knowing, without seeing, without looking at that, if that's what it genuinely says, it wouldn't worry me. It's a nice little coincidence. It wouldn't worry me too much. Matt, is there anything you said that you want me to rebuke? I have got lots of things written down here, but while you're here, let's, if there's specific things, I'll offer my response.
Oh no, it's okay. Wait, I just go through your notes. We'll probably get to everything. Maybe.
Donny, is there anything else?
And we'll have time at the end of our closings to ask each other some questions that we feel maybe we did not answer well. And so, Catz, maybe take a few of what you wrote about Matt's arguments, and then take your final minutes addressing that. And then if you got time, feel free to go through your other slides too, Catz, because you still got. Let me see, 17. So you still got 8 minutes.
So I wrote some things here. In fact, I put that there because that's terrible. The grammar on that is terrible. I didn't bother with my spelling because I wasn't going to put it on screen. Matt, one of the things I've got written down from him is, with DNA repair, we can live to 1,000 years. Now, to me, that's another, well, what if? What if we used to live to 1,000 years? And it will be nice if CRISPR come along, and I know CRISPR is extending the life of mice and etc. But when we look back, and any of the genetic analysis that we're doing on, trying to trace back, for example, to mitochondrial Eve, or we're trying to trace back to any of our ancestors, there's just nothing there. It's like, wouldn't it be nice if I could open my drawer and there was £1 million in there? That would be lovely. Oh, that would be great. But that's not the same as saying, wouldn't it, I believe that at one point in time, there was £1 million in my drawer. So it is a belief. And that's all it is. It's a belief. Now, the thing that I found a little bit was when you were talking about bone structure and asking if people like Homo erectus, etc., could live, much longer than us. Well, again, when we look at Homo erectus and we try and estimate from the fossils how old they lived, we look at, one thing we'd look at is the size of the body, the development. Another thing we'd look at is the wear and tear on the teeth to get an estimate of their age. Similar to how we might, if we found like a burned body today at a murder scene. Obviously, we've got genetic analysis, but, so maybe not today, but throw us back 50 years, similar to how we might estimate ages then. And there's just no suggestion anybody has ever said, our species like Homo erectus, whatever, have lived longer than modern humans. It's just none. So I know you brought up the tortoise and you said, well, the tortoise has got thicker bones, but then again, it's got an entirely different physiology. So I don't think we can say a tortoise lives for a very long time because it's got thicker bones. I mean, a tree can live for thousands of years, and that's not got thick bones, so we're going to say Noah was made of wood. There's no real link there for me. Now, you talked about the Eve consensus sequence. I'd like to ask you this later on, because at the time I was typing away a rebuttal for something else, and that kind of passed me by. But as far as I know, the Eve consensus sequence is mitochondrial DNA. And people do differ in the Eve consensus sequence. I got the impression at the time you were saying they didn't differ. Maybe I've completely botched that up. I only caught like half of it because I was trying to write something else, and apologies if I botched that up. But I got the feeling you were saying they didn't differ, but actually they do. But it's not nuclear DNA, it's mitochondrial DNA. So I'd like you to maybe flesh that out later on if possible. Now, you did come out with a fantastic quote. You said, all predictions about Neanderthals have been wrong. And I like that. But I would like it if there were specifics. What specific prediction and what was the context? So for example, my daughter, she's out tonight with some friends. She's just about to turn 18. We're given a slightly longer leash. And I can make predictions about which friends she's out with, educated predictions that I'm prepared to be wrong about when I find out in the future. Is that what happened? Is that what you're referring to? That historically, when we've started researching and finding Neanderthal bones that we've made educated guesses that we're not sure about, and then evidence has showed us it's something else. Well, that's not really a problem, is it? You know, I could think she's out with her friend Claire, but actually she's out with her friend Penny. Okay, doesn't make me an idiot. Doesn't make me, you know, it's which predictions exactly? I'd really just like specifics. What I've tried to do is bring the names of the papers and the quotes of the papers I'm using. And I'm sure you're very well researched. Let me just have a look. But to me, it would have been helpful if I'd like the names and the quotes from the papers to get some context for that. You talked about mutation rates, which I think I've addressed mutation rates, specifically the mitochondrial mutation rates. We talked about the bottlenecking because of the ureagenesis bottleneck, and also the mutation rates with the Y chromosome, which would all be expected. And what I put down here is, to me, it's interesting that when you're talking about mutation rates, both of you actually, the only mutation rates that really you're bringing forward as evidence for your stance are the mitochondrial mutation rates and the Y chromosome mutation rates, not nuclear mutation rates. And that to me is interesting because both the Y chromosome and the mitochondrial mutation rates are very, very easily explained. Like we said, there is a high mitochondrial DNA mutation rate, but then there's a bottleneck every time an egg is formed. So that mutation rate won't actually lead to huge diversity that you might think it might do as offspring are given. And with the Y chromosome, there's no recombination to wipe out mutations. And when you get billions and billions of sperm formed in a single ejaculate, obviously you're going to have high chances of mutation with the sperm that fertilizes the egg. So to me, they are absolutely what I would expect to see with the mitochondria and the Y chromosome. And it's interesting to me that neither of you talked about nuclear DNA mutation rates. Now, I don't know how long I've got left. Could you just, anybody? I'll do a dance. I'll find out. Okay, I don't know how long. I'm just going to keep going till I find out. Okay, we've got, what should we... complex system. Why should we believe in complex evolved systems was one of the questions I was asked. I think it was you, Donny, that asked me this one. Well, we look at, like I said at the very beginning of my presentation, we look at the evidence and we see whether the scientific evidence we can find will disprove our ideas. And it isn't disproving our ideas. My question, obviously, would be the same to be thrown back at you. Why should I believe in a talking snake when there's been no research? Why should I believe in dust that contains no nucleotides, no sugar, no protein? Why should I believe in that becoming a human? So I think we're on a similar par with that. You know, you might not believe in evolution, and it's fine if you don't believe in evolution. But it goes
Catz, you have 30 seconds, just to let you know.
Okay, thanks. And oh yeah, the junk DNA. Why do we have this junk DNA? We have these extra bases. It places an extra energy demand on us. Well, actually, so does a mole on the skin, and does that drop off? Do you know what I mean? Yes, we get extra bases. The energy band will be tiny, 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 and insignificant. You know, it's nothing more than a little spot or a mole. Why do we have a spot or a mole? Why do we have a mutation that gives us an extra base? It's not really a knockout punch.
Okay, Catz. Appreciate it. I gave you a little bit extra time just because you were gracious with your rebuttal by asking us a couple clarifying questions. And so, okay, we're moving along smoothly, and we're in our closing sessions now, our summary sessions, which means we will have some pretty good time to engage the audience and then ask each other a couple of our own clarifying questions. So Catz, we're going to put you backstage. you can relax and get yourself ready for your closing. And closings or summaries, we have 12 minutes. And so we'll break it up about 6 minutes each. Matt, I have a lot to cover, brother. So I am going to go fast for 6 minutes. And then once I get to about the 6 minute mark, I'll hand it to you. And okay, let's start the timer. Am I coming in smooth, Matt? I was getting a bit of an internet signal there, but
sounds good.
it's coming in smooth. Okay, good.
No, it's good.
Okay, here we go. So Catz, new points. I'm pretty satisfied with how we took care of his opening statement, addressed the major points, the most prominent ones, I believe, and still touched on the dating methods, coal, some of the other objections more so related to the flood and age of the earth. And so, okay, these are his points from his last rebuttal. And first thing I want to address, though, I think this is an important issue, is this would be a really, really major coincidence for the evolutionists that it just so turns out that all mitochondrial DNA worldwide can be traced back to 3 main nodes. Now, it's more of evolutionary philosophy or an evolutionary assumption to say, well, these have to be populations, or even Eve has to be, have been in a population. These bottlenecks don't work. Okay, the out of Africa bottlenecks, that would have comprised in the evolutionary model severe inbreeding. Okay, so I'm not sure Catz really addressed my point about the low diversity that's expected from a biblical model, but for the evolutionary model, we should expect higher levels of genetic diversity. Why are there so few erectus? Where's evidence in the fossil record for the hominin story? I'm not sure we got a decent answer to that one. But again, the evolutionary rescue device that, well, the reason why we even have this lucky man, lucky woman is because of bottlenecks and smaller populations over time and inbreeding. And so we're left with just coincidentally or mathematically with 1 woman, 1 man, even though the Bible demanded this. Our resolution tonight is Adam and Eve are real. Biblical creation is true. It turns out that the data has discovered Adam and Eve and confirmed biblical origins, but the evolutionists, they got these post hoc, ad hoc rescue devices after the fact. Okay, where did they predict this data? And the out of Africa population bottleneck, okay, so humans are reduced to, let's say, between 2 and 10,000, between 70 and 200,000 years ago. Well, that would be necessary to reduce levels of heterozygosity and increase levels of homozygosity. Problem is, that would have to be a prolonged bottleneck. What that means is you'd have severe inbreeding. You'd have all these deleterious mutations that have accumulated. They're in recessive form. Now they're coming to the forefront, leading to rapid genetic degeneration. They're causing disease. Okay, because inbreeding reveals the hidden reservoir of genetic mistakes over time. This isn't a 1-generation bottleneck followed up by rapid and exponential population growth like our bottlenecks. The creation bottleneck, the flood bottleneck, and in many ways what happened at Babel is not an issue for us. It's 1 generation followed up by rapid population growth. But the evolutionary bottleneck, it's not feasible. This population of between 2 and 10,000, some say 30,000, they would have been so incredibly genetically compromised that there's no feasible way. This is evolutionary philosophy. How would they have spread out to all parts of the globe, seize dominion over the planet, invent the space shuttle, invent computers? So it's the evolutionary story that says, well, Adam and Eve, they had to have belonged to a population. Now, in many ways with the Y chromosome, yes, our last Y chromosomal ancestor is Noah, going right back to the flood bottleneck, who could have been quite similar to Adam, depending on how many mutations accumulated. Catz said we didn't speak about autosomal nuclear DNA. Yes, I did. The mutation rate in the nuclear DNA is high, 100 new mutations per person per generation. That puts a genetic burden upon species. That puts shelf lives on species. If they cannot persist for millions of years into the future, they could not have persisted for millions of years into the past, which refutes the fossil record because we have biology represented in the fossil record through fossils, extinct creatures. And the fast neutral mutation rate equals or approximates the fixation rate, according to Fatuma. So The bottlenecks don't work. Those are rescue devices. This doesn't work. LM and N. Notice this. I really want people to see this. They're separated by short lines. Matt has a visual. He looked up how many DNA differences separate the three, and they're only a few, less than 10. So yes, these could be 3 individuals, and radiating out from those nodes are long branches. That suggests a lot of DNA diversity, which makes perfect sense because the pre-flood world would have been less time. People were living longer. There'd be a lot of overlap. And so L, M, and N could have lived at the same time. I'm unsatisfied with the way Catz has addressed this. This is exactly what we would expect. And Catz says, well, that's a nice coincidence. But that is a huge coincidence. To a biblical creationist, this is exactly what we'd expect. Genesis 9:19. These are the 3 sons of Noah, and of them was the whole earth overspread. Mainly created units of DNA function. Yes, the majority of ERV sequences are not actually true ERVs. They represent solo LTRs, and these solo LTRs serve as binding sites for initiating or enhancing gene expression. So yes, if we share all of those LTRs with other creatures like chimpanzees, then certainly they can be created units of DNA function. How does Catz explain the origin or evolution of those functions? It's Catz that assumes those solo LTRs were once a full ERV. Okay, I've got an entire presentation going over the structural components of ERVs, every single component, and how they act in the function of these ERVs. Notice this right from the secular literature. These single LTR promoters, they have a role in regulating cell immune response. These ERVs have to have that structure, the GAG, the pole, the ENV components, the LTRs. And here's the thing. They like to say, well, it's just a couple that are functional. No, we have 80% genomic activity. We have evidence for that. This has to be legit activity because this activity and transcription in general is an energy intensive, resource intensive process. Every time you add a single nucleotide to a growing RNA chain, you're consuming energy. If this was junk, evolutionary mechanism should have weeded it out over time. Within that activity, we find pseudogenes, ALU sequences, ERVs. That suggests that they are functional. I have one paper that says if we didn't have ERVs, we couldn't exist because they're functional in embryological development. Notice this. Long disregarded as junk DNA or genomic dark matter, ERVs turned out to represent important components of the antiviral immune response. They're also functional in tumor suppression, where transcribed ERVs give the cell the appearance of being invaded by a virus, and therefore the immune system, acting with the guardian of the genome, the p53 protein, targets that tumor cell with destruction. But guess what? The ERV has to look like a virus in order to carry out that function. It's the same thing with their functions in fighting off exogenous retroviruses. They require the GAG, the pole, the ENV, the LTRs. They require those. So every single component of these amazingly designed DNA units are functional. There's so much to discuss on ERVs. It's just a small portion of this debate. The evidence for Adam and Eve, which is our burden tonight, being in the affirmative, that should be the main focus. And currently I really like Catz. I'm unsatisfied with the way he's rebutted that. And so perhaps a longer informal discussion one day, Catz and I can discuss ERVs. Okay, Matt, you have 4 minutes. Go ahead, brother.
Right. No, a snake did not talk. So there you go, Catz. No, it's that was actually the devil in the book of Revelation. It mentions that he's that old serpent, referring to him as the serpent that was eaten, that tempted Eve. I mean, think about it. If a snake started talking, would you really trust it anyway? Probably not. But if it was an angel, you would be much more prone to believe something like that. Homo erectus and heidelbergensis, we're not saying that they lived for 1,000 years because they're living on this side of the bottleneck. We just said that they lived longer lives, and that's why they have that propensity inside of their bones to show that. And I showed that it was genetically possible, even with people today, reverting their genome. I also showed that species that are related to one another do live longer based on their bone thickness. So other than having an ID on their body, I don't know what else I can do for you. You wanted Neanderthal failed predictions. I actually have those. So they were considered dim-witted brutes. One of their predictions was called Homo stupidus. They were too dull and clumsy to use tools. They were subhuman. They wouldn't ever mate with people. They also said that they wouldn't predicted to be found in African populations. Their DNA has been found in them. They were scavengers. They were incapable of language and thought. They were not intelligent. They weren't able to use a wide variety or adapt to a wide variety of ecological zones. So all that has been, I just had that on the top of my hands. Good thing I did. You wanted evidence. You also said that Parsons actually didn't publish that date. He actually did. We can see it right down here. That is directly in his study. And he said that the rate was about 20 times higher than expected. So he actually did say that. And the secular paper that came back after saying that this, with the new clock, she would be 6,000 years old. That's also secular. This isn't young earth creation stuff. They're literally admitting like, uh-oh, this isn't good. But it doesn't go back just then. We can go into the future. In 2005, we found the exact same substitution and mutation rate, same as Parsons, 1 mutation, 29 generations. Then we also find it in 2012, Parsons mutation rate, yet again, 13 to 42 times higher than the phylogenetic evolutionary mutation rates. And then we can go to this. Deep-rooted pedigrees go back 200 to 400 years. In all these studies, fast mutation rate every single time. How about recently, 2022, Jasmine, 1 substitution every 38 generations. Same, 16 times faster than the evolutionary phylogenetic mutation rate. But let's keep going. As in other studies, our estimates are much higher than the obtained phylogenetic mutation rate. Jensen has the same thing. Here's how he did his calculations. They tried to say, well, maybe like you said earlier, maybe they counted somatic mutations, or maybe they were counting different heteroplasmy because there's different mitochondrial passed on. The more mitochondrial that gets passed on, the worse it gets. It speeds that mutation rate clock up times 4. So if 6,000 years old is bad, it's going to get even faster, adding heteroplasmy. But what did they actually say? Here he is. He ran the numbers and said, it's rather the phylogenetic rate estimate that is biased. Oh, interesting. The biased one is the evolutionary one. It's clear that the pedigree mutation rates provide the direct estimate of the mutation rate, exactly what we've been saying. So if these pedigree rates are so pointless, why do they keep using them? Why do they keep doing studies? And they match real world things. And they also match genealogies. This guy ran a simulation, 2 different models to go back to the most recent common ancestor in genealogical. And it matched exactly the same thing. His was different, though. He found the most recent common ancestor ended in East Asia, not Africa. So notice that the fast mutation rate and substitution rate land on the maximum diversity we see in life today based on the young earth creation timeline. We can explain it. That's all we need to do. That's it. It's in the peer-reviewed literature. Sorry, what?
10 seconds. But if feel free to wrap. I know it flies by. Wrap up your thoughts.
10 seconds. Okay. Sure. They said this paradox is long-standing. They can't figure out how to get through catastrophic bottlenecks. They tried to say, well, maybe if we have one every 50 years, it can be true. Think about that. Impossible. That's regarding a population growth rate of 0, which they need to have humans living in Africa for hundreds and thousands of years. They need 0 population growth rate. So they had to invoke catastrophic bottlenecks.
Okay, let me make sure we're on track. Gentlemen, this has been great. I'm impressed with both of your endurance. 3 hours doesn't really feel like 3 hours. A lot of presentations here tonight. These are important that we get good representations of both sides. And I appreciate that Catz has put in the prep.
Do we have like 20 questions?
Oh, well, we definitely won't be able to get through all the questions, but we'll, and we still got Catz. We still got Catz closing statement. Okay. But to the audience, anybody just joining us, we're moving along smoothly in this seminar. We just hit the 3-hour mark roughly. We've had 50, not 15, 50-minute opening statements, lots of points and arguments made. We had 25-minute rebuttals or responses. We're now in the 12-minute closing statement section. So Matt and I just went about 12 minutes and 20-ish seconds. So Catz, to be fair, and because we love you, you also get 12 and a half minutes. And just please let me know when you're ready to go. Click on mute, and you got 12 minutes. I can put your slides up as well if you'd like to, Catz.
Let's go. Okay, I really enjoyed it. Thank you for having us, both of you. It's been a pleasure. What I would say is, Donny did say in his summary, he was a bit unsatisfied with some of the rebuttals I gave, which is fair enough. There's a lot of information and there's a lot to respond to. And it probably is a good idea to maybe do a follow-up where we dig into some stuff a little bit more deeply. I feel the same the other way around as well. You know, I've thrown a lot of stuff and I've been probably my mind wanting answers to particular things. But of course, I've not asked for those. I've not dictated that. And I've got answers to other things. So there are some things that I feel I would like more in-depth answers to. So I'm just going to go through a few of those. One of the things I've heard a lot is deleterious mutation, deleterious mutation, deleterious mutation. Now, one thing I brought up was this E. coli long-term evolution experiment, something that we can observe, and we have been observing. Obviously, some mutations are deleterious, but some are beneficial, and some do nothing, and some deleterious ones, because of selection, just get removed. You know, and if it's very deleterious, then we die before we can have offspring. So the idea that a longer a population goes, they pick up more mutations, that has to be a bad thing, isn't right. And I showed this paper here, the E. coli long-term evolution experiment. And we showed here that, you know, in this experiment, most of the mutations that are being passed on down the generation are advantageous. It doesn't mean that the vast majority of mutations are being passed on. Maybe there are deleterious ones which are being removed, but actually, as a whole, the population is benefiting. These bacteria are getting bigger, they're getting more resistant. You know, they are becoming far more suited to their environment. You know, and again, creationists don't deny selection. They don't deny what they will call microevolution. So in that sense, they do agree that these mutations and, you know, lead to adaptation. So it's not, they don't even claim in that sense that they are deleterious. So I don't know. I'd like to touch on that a little bit, because there are plenty of examples of mutations being observed that are actually beneficial. We talked about antibiotic pumps as well, which wasn't kind of referred back to in the rebuttal. Obviously, there's lots to get through, but surely these are advantageous structures that are forming, pumps that can pump out an antibiotic from a bacteria, or literally changing the structure of your ribosomes. This is obviously an advantageous thing to do. It's obviously new information. So it would be nice to hear what the rebuttal is to that in a future discussion. You know, why is this kind of overlooked? Why do we have to talk about mutations as being deleterious all of the time, every time we talk about mutations, when actually, obviously, we both agree, at least on microevolution, where we are getting benefits, and we observe in the real world now, over years, we observe bacteria in the lab forming these structures. So that will be interesting. Another thing I would like to have touched on, one of the things Matt said in his rebuttal, if I can just find it here, was everything is linked. Everything is linked to radioactive dating. Well, if I look at what I said here before, I deliberately went out my way to show things that were not linked to radioactive dating. I showed the pyramid. We talked about the Khufu ships that hadn't been carbon dated. And we talked about dating things around them in the pyramid using, for example, thermoluminescence. And if you wind back the stream, I really stressed nothing to do with radioisotopes, nothing to do with them at all. Dendrochronology, again, I stressed absolutely nothing to do at all with radioisotopes. And yet they are bringing back the same results to date the furniture and the pottery that was in the room that the Khufu ship was in. So this kind of, to me, it's just a very cheap hand wave dismissal to say, well, actually, radioactive dating is flawed and everything is kind of linked on that, and we can just ignore it. Well, we can't. To me, that is, it's not really addressing the situation. It's running kind of from the situation, really, will be my honest stance. So it'd be nice to talk about that in the future. Something else that wasn't touched on at all, which I would really like to address, was this study here, where we said 57 different studies across the globe, 57 spanning decades, that were looking and using different techniques to date, to look at the temperature at different dates in the past, all came up with, essentially, when we plot it on a graph, obviously some of them could date further back than others. But when we look at the first few 100,000 years, which was here, we're getting exactly the same results from studies decades apart. And again, I asked Ray Comfort this, and he was honest. He just said, I don't know. I don't know. And I understand when I present this to people like I've done tonight for the first time, like I was before with that paper thing. I need, with the mitochondria and the 3 different nodes, I need to read the paper. If it's coming from a scientific paper, I need to read it, right? So I understand that Matt and Donny will need to read this and try and really get their heads around why they are all giving the same result. But that will be a really nice thing to have a chat on in the future, because to me, there is no explanation. How can you have different dating techniques and they all be wrong, but somehow they are wrong in a way that makes them all agree with each other in this kind of manner? And we saw the same thing with the Vostok. Where are we looking? I can't find it. Anyway, we saw the same thing with the other study with the 2 locations in Antarctica. So to me, those things will be really, really nice to be addressed. I do believe I've got a few minutes left. So again, with isochron dating, I would actually love to see the graph that Matt showed before. He was talking about concordance, and to me, it looked like he was taking different isotopes from different rocks and then saying that the rocks were a different age, but they were different rocks. I might be completely wrong, but I'd like to see that again and go for it. If they were all the same rock, then actually what you were seeing were error bars overlapping. And if they were the same rock and the error bars were overlapping, then they're in the same ballpark because the error bars are overlapping. That's no problem. They were still dating them as being hundreds of millions of years old, not 6,000 years old. I don't understand where the problem is. So I'd like to have a chat about that in the future, because to me, there's absolutely no problem. But what I'd really like to do most of all, to be honest, is in the comment section of this video, because I know on this channel, most people will be creationists and they'll be like, oh, the evolutionists got destroyed, and because that's what it's like. And on the 810 Catz channel, it'll be the other way around. But I would love to see creationists say, okay, yeah, this here, right, is just a belief. I just believe that God turned dust. That's my get out of jail free card, dust into a man. I do believe, as a get out of jail free card, that when the Grand Canyon was formed, the topography was formed because God decided to quickly raise up the mountains and drop the ocean basins. It's just a belief. There's no scientific evidence to say that kind of geological process has ever happened, can ever happen, or is happening. I just believe it did, because there is no evidence that has ever been presented to anybody to show that that kind of geological process at those kind of rates has ever happened or will ever happen. So to me, that would be nice to see. How long have I got left? 4 minutes. I might do another dance. I'm just trying to think of what else I'd like to see. I might call this in a minute if I can't think of what my last argument was. I did have a couple, but Donny's come back. Have you got any questions or?
Oh, well, I was just going to say we do have until, we got about another 25 minutes, so we got to wrap up. So feel free to end your closing anytime, Catz. I don't want you to feel like you have to.
Yeah, I'll end it there. I'll end it there.
Yeah. Okay. That was good. I got to say, Catz, you're a good sport. You're a good sport. And, there's only so much you can address on both sides in, a debate seminar where we're both giving 50 minutes. And so I would say you did a good job addressing what you could. Hopefully we did a good job addressing what we could, but there are a couple things that we did not necessarily get a full opportunity to engage, which is why I believe utilizing this next 25 minutes, Catz and Matt, could really help make this seminar that much more comprehensive. And so why don't we devote half of it to addressing just a few of these audience questions? Leave it up to me to pick the super chats. And then let's hold each other's feet to the fire on a couple issues. Catz, perhaps you'd want to ask us about thermoluminescence dating or the ice cores or something like that. And we can ask you a couple things. So if you think it's fair, Catz, you just ended with your closing. So why don't we get a couple questions in first, and then you throw a couple questions at us.
Sounds good.
You're a good sport, Catz. That's why, like even after almost 3.5 hours, you're still our favorite evolutionist. Rue Hef, $10 super chat. Hey, Donny and Matt, when are we debating chromosome fusion? Okay, good question. I did bring that up in my opening just briefly with the article that I wrote. And so, Ruhiv, perhaps we can engage in some kind of debate seminar or discussion as well, as I know that's your specialty. Another question real quick. Are super chats blocked? No, you should be able to send in a super chat, as we do have some. Let me double check. Yeah, it should be available. But okay, let's utilize our time here, because Catz, we do want to respect the time you've given. So glad you agreed to this. Been a ton of fun. And so we want to give you the opportunity to ask a couple questions too. Can you reiterate your response here to the Y chromosome dissimilarity argument, Catz, where I pointed to the Y chromosomes, but also specifically the latest data on it published in 2024, where now the human Y, you can compare all the different grade 8. Humans are considered a grade 8, human grade 8 by common descent proponents. And so you bring it up to the chimpanzee Y, and it's only about 26.2% the same. In my rebuttal, I put down a few different points, one of them being the nature of recombination. You'd mentioned recombination and how recombination is not present for the most part in the Y chromosome, right? You got your uniparentally DNA inherited compartments, Y chromosomes inherited paternally. There's really no recombination. Some say at the tips, or maybe there's a little bit of gene conversion, but the point is really no recombination, which means I would argue that the Y chromosome should be relatively more stable, because it's not like autosomal DNA where you have genetic shuffling every generation of these genetic variants that leads to new chromosomal combinations. And so that's why Y chromosome, mitochondrial DNA, they're good chromosomes to build family trees and work your way backwards to last common ancestors, both a male and a female. So I did take note of that. So I guess 2 questions, and then take your time, Catz, because I appreciate this. Question 1, can you reiterate your response as to why, according to evolution, the chimp and human Ys are so different? And 2, why do you believe recombination would help you with that? Go ahead.
Okay, so they are so different. I mean, recombination is a big part of the answer, but let's look at when mutation occurs, right? Mutation occurs during cell division. Now, when I keep saying the word ejaculate. It's the most I've said that word in a day for like ever. But when the testicles are producing an ejaculate, I'll say it again, there are literally billions of sperm, aren't there, present? And half of those will carry the Y chromosome. So half of billions is still billions, which gives billions of opportunities for mutation to occur. It's a very, 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 very rapid process. And then you've got those sperm racing towards the egg. And you're going to have this fantastic kind of like diversity across the sperm because of this mutation, because of this huge amount of sperm that have been produced. So when one of them enters the egg, the chance of it having mutation is going to be quite high because of the sheer number of sperm that have been produced. So it's not a surprise that then the offspring will have a slightly different Y chromosome, and then we go on and on and on and on and on. When it comes to recombination,
actually, Catz, before you continue, because I do appreciate those points, but I'm wondering why then, that being the case, why in humans, okay, the Y chromosome, even if you were to assume evolution's true and we take Y chromosome atom back, 100 to 200,000 years into the out of Africa scenario, in all that time, because there's good work now being done on Y chromosome mutation rates that suggest the mutation rate is somewhere between 1 and 3 per generation, which is fast. The amount of DNA diversity in the Y chromosome in men is on average probably about 200, and the higher limit might be 500-600. Okay, those are not exact numbers, but something like that. The Y chromosome is a larger chromosome. It's about 65 million letters long. Mitochondrial DNA is about 16,500. Okay, so if what you're saying is true, which I'm not saying it isn't true, but for this to account for this dissimilarity, why humans? The human male Y chromosome, every single Y chromosome on the planet is nearly identical, even with the most divergent groups, people that have been isolated for, you could even say, hundreds of years, they've got a very similar Y chromosome. And so if what you're saying can account for the dissimilarity, why are human Y chromosomes still so vastly similar? How come they haven't diverged more in 200,000 years, Catz?
What, human Y chromosomes similar to
each other?
Yeah,
every single human Y is nearly very low diversity, very similar.
Well, there are actually significant differences in structure, maybe genetically. And the reason that genetically the diversity wouldn't be as great is because obviously if the genetic diversity was big, then it wouldn't function. So it's not, you're not going to get viable offspring. But actually there is, if you look at structure and size, there is considerable diversity between in structure and size in the male Y chromosome. So there actually really is.
Anything similar to that? Because you'll notice on this diagram, Catz, the chimpanzee Y, it lost the entirety of its heterochromatic arm from your model. When did, because humans, okay, maybe there's some, intravariation or some differences there, but it's not like there's any human that lost the entirety of its heterochromatic arm or lost half of its. I mean, these are literally half the size. And so according to your model, when did this happen? When did this entire heterochromatic arm, when was that lost after the split?
Oh, I have no idea when that would have been lost. No idea. I haven't looked into the dates or stuff for that. But what I can say is there is huge diversity in size and structure due to these large non-coding regions. But that's well documented. And I mean, maybe while we're talking, Matt can Google it and check that I'm not pulling the wool over your eyes or something. But give it a Google. And there really is, or anybody watching at home, there really is massive differences in size and structure.
But would you agree that regardless of the differences, whatever those be, either minute or somewhat larger than expected in humans, you would agree that all human male Y chromosomes still have very low variation, 99% the same?
Are we talking genetic variation? Coding variation or non-coding?
The Y chromosome, the entirety of it, gene content, architecture, size differences, everything.
Well, not size differences. Like I said, they are significant. Structure and size, it does change it. But you got to remember, we have. So you saying, if you're saying that there's not much diversity when it comes to the genes, well, that would make sense. We've got the reproductive organ here, haven't we? The reproductive chromosome, and quite a, those genes are obviously extremely important if it comes to propagating offspring. So mutations would have to be beneficial in those genes, otherwise you're not going to get a viable offspring, in which case those sperm wouldn't work, they wouldn't fertilize the egg. So it's not that these mutations aren't.
But what if they're neutral, like nearly?
If they were absolutely neutral, they'd probably be occurring in the non-coding regions, wouldn't they? They'd either be beneficial or if they were neutral inside a coding region, you wouldn't notice them, would you?
Okay, so here's my next question, then feel free to answer the recombination one. Do you, because you would concede at the moment that you're not sure how the heterochromatic arm was lost, or I would also assume that, so for example, there's data suggesting that the chimpanzee Y chromosome has twice as many large palindrome sequences as the human Y. So if the chimpanzee has twice as many of these kinds of sequences, which are used to protect important genes in male fertility, and the chimpanzee's lost most of its heterochromatin, okay,
Mhm.
And would you agree that you don't really know when that happened or how that happened?
I have got no what in terms of
Yeah, well, I'm just
whereabouts
saying,
in evolutionary history, it's got to be after we've diverged, doesn't it? It's got to
right.
be after
So
we've diverged.
in order for you to hold the idea that, okay, these changes even took place, wouldn't you or an evolutionary scientist in the field, wouldn't they have to provide a sophisticated mathematical model that actually shows, hey, this is how, this is where, this is when, this is why all this heterochromatin was lost, or this is how these palindrome sequences were added or lost, depending on what view you take. In order for this to be science, wouldn't there have to be a model for that rather than just an assertion like, well, maybe this, maybe that? Katz, I'm curious to see your thoughts on that.
Well, the mechanisms for deletions and replications of genetic material are well known. I mean, you know that yourself. It's not there's no query about the mechanisms that lead to DNA loss or the mechanisms that lead to replications of base sequences and stuff. So it's not about the mechanisms. When you're talking about an absolute date, I mean, it's something I've never looked at, but obviously it will have happened after we split from our latest
Right.
common ancestor.
It would if evolution is true, it would have to, right?
It would have to be, yeah, I would imagine.
But why the loss of the entirety of the heterochromatic arm, but it's very present in humans? Or why the addition of palindrome sequences? Or it seems like more okay, you said a lot in your presentation, like, well, we creationists, we have to believe certain things. And I would argue that on certain issues like this, you have to believe that these changes took place. You, in a way, you're extrapolating. You're saying, well, today we know that deletions happen. We know that duplications happen. We know that other genetic mechanisms happen. Therefore, we're going to extrapolate that to mean that these must have happened in abundance at some point after the human chimp split. But I would argue that's an extrapolation fallacy. That's more evolutionary philosophy rather than empirical science. Katz, what are your thoughts?
Well, my thoughts twofold is it would fit everything else. It would fit everything else that we have in place. All the other genetic analysis, all the mitochondrial DNA analysis, all the fossil records. It would fit that absolutely perfectly. So it's not a case that it's a long shot. If you look at any one thing in isolation, you can say that, but obviously it would fit really well. My second point to that would be, I mean, we all know here that the heterochromic section of the DNA really is very largely non-coding. So if it gets lost, will that Y chromosome still be functional, and will it promote viable offspring? Well, yes. So there's no reason that it can't be lost. There's no reason that through mutation that actually is in any way a problem. You know what I mean? If it was a big coding part, then it might be an issue. But it's not a coding part at all.
But even in non-coding DNA, there's incredible functions in regulating the coding DNA in terms of heterochromatin that protects genome integrity. It maintains the structure of very specific chromosomal regions. And so I think it's more of an assertion to say, well, because maybe it's not as important as other genes or genetic sequences, it could have been lost and tolerated in the chimpanzee line. But then why is it so prevalent in the human line, Katz?
Well, why would it have to be removed if it's not deleterious? If it's I mean, I said before.
Are you saying in the chimp? Are you saying in the chimp it was present or not present?
No, I'm saying what I thought you said. Why is it there? Is in like,
well, yeah. How come it's there in the human line but not there in the? So obviously it was lost in one lineage.
Yeah,
but it was retained in the other. Why?
Or was it duplicated in one lineage and just not there in the other in present?
Right. And here's my point. Aren't we now kind of going into the world of what if this, what if that? Maybe this story works. But I want to, how is this empirical? How is this?
What I'm wondering is why are you sorry. Why would you expect them to be exactly the same? Why would you expect them to be? That's curious to be like.
Okay, well,
that sounds like what you're saying.
Yeah, no, it's a great question. I would expect them to be a lot more similar than 26%. I mean, if we share a common ancestor with the chimpanzee and go back 6 million years to a common ancestor, and yet all human Y chromosomes are 99% the same, but less than 30% with the chimp, well, you'd expect a little more similarity than 26. I'll say this, and then you can respond. I see you're sharing your slide. I mentioned in my opening, possibly my rebuttal, it's the differences that make all the difference, right? We can both explain similarities. I would say similarities are due to common design. You would say similarities due to common descent. But it's the differences that make all the difference. Yeah, the evolutionists can explain some differences, but it's once we get to these massive differences where you're looking at, 70% difference between supposedly our closest cousin, that's where I would say the burden of proof is on the evolutionist. Katz, I said a lot there. Go ahead, take your time.
Can I just share my screen? Is that okay?
Yes. Yeah.
I feel like a lot is being made out here that the structural difference is actually a huge difference. But I had this on my PowerPoint, which I didn't show before. We can see the 2 chromosomes, and obviously this is a heterochromic bit for humans. Nice little graph here to actually show what the difference is. Now, in the ampliconic section, which is basically the bit where we contain multiple genes over and over and over and over and over again, there is only about 50. With other species, there is obviously a different. This is how we compare, with other species, right? With the X degenerate chromosome, we compare very well with other species, but in the Y chromosome, we don't compare too well with other species, but we are not that different from each other, is the point. I'm not wording this very well, but the way we compare with other species, although we're quite different from them, there's only you're talking as if the difference is huge.
Well, it is huge
here. But is that what's being shown here? I mean, I'm seeing a few percent difference when it comes to the ampliconic.
But you got to look at you got to look at the gene content, the size differences, we got to look at the whole picture. And when you consider overall gene content, size differences, and architecture, which also includes that heterochromatin and the palindrome sequences, that's where we find that overall the Y chromosomes are less than 30% the same.
And if you are talking about looking at the whole picture, then let's look at the whole picture. And the whole picture is that our DNA is 98%.
Okay, but Katz, in my presentation, I did show that paper from 2025, just a couple months ago, the sophisticated sequencing data of the great apes. And in the supplementary material, they noted that 12% of the human and chimpanzee genomes don't even align. What are your thoughts on that?
Align in terms of what? Basics, or when we're...
Yeah, like there's whole set. So gap differences. So you could take the chimpanzee genome and the human genome, and there's certain regions in the chimp genome that have no alignable counterpart to the human genome and vice versa. So there's sections that don't even align. So you basically have to discard them and say, well, okay, there's no way to align these, so we'll just compare the regions that do align.
But of course, you do know that if everything aligned perfectly and everything is the same, we would be the same species.
Right, but why 12%?
Well, if it was 11% or 11.5%, you could say why specifically 11%, 11.5%. We obviously are different.
Well, would you agree? It's certainly more than 98% different. Think about it, if 12% of it doesn't align, how can you say it's 98% the same?
But when you say align, are you talking about the base sequences in coding regions or non-coding regions? Are you talking structurally? It's quite a generic question. Like if you could focus in on.
Yeah. Oh yeah. It could be an entire sequence with a whole number of base pairs. Let's just say it was 100,000 base pairs, and I'm just throwing a number out there. You'd have to read the paper. Let's say those don't align. Well, an evolutionist could say, yeah, that's because there was a massive deletion of 100,000 base pairs or something like that. But so yeah, there's a lot there that's simply not. But here's the thing. Humans and chimps overall are still the closest. There still is that hierarchy. But when we focus in on, and I'll say this because I do want to give you the chance to ask your question, Katz, but when we focus on the differences that poses a challenge, because Katz, you'd have to explain how can those differences arise? How do they become fixed? so, you know, so on and so forth. And so basically all I'm asking for is a model, a mathematical model. When did these changes take place? Why did they take place? How did they take place? Would you say that's fair? And then feel free to have the last word, Catz.
Depends on, I mean, when you say mathematical model, it depends on the detail. You know, when you say when do they take place, are you after like a calendar year? I mean, or you after like a location on the ancestor? It's
1 million BC.
Yeah. I mean, are you after like a specific point on the evolutionary tree?
Well, for example, I'll say this. Maybe this helps clarify. Let's say with the human chromosome 2, for example. Let's assume that took place. It seems more of like just a general argument rather than an argument with substance. Because in your opinion, if there was a fusion, because that would be a difference, right? We have 46, apes have 48. When did that take place? Did that take place in an Australopithecine? Did that take place in Homo habilis? Because you guys just say, well, this took place, but then you start getting into the details and it's usually, well, I don't know.
I am absolutely positive that that this would be something we could Google now and find. Like my presentation tonight was really on, obviously we didn't touch chromosome 2, but
right.
Really
and I wasn't saying that to get into a whole debate.
Oh, I know.
I'm just assuming if there was a fusion, it'd be interesting to know when it happened and what lineage it happened, because you'd have to agree that would have been a near extinction event, right? Whoever got that fusion, they're basically the ancestor of all people today, because all people today have 46 chromosomes, which means that fusion event would have had to become fixed, and all other lineages that have 48 would have had to been wiped out. Right. So there's a lot of details there basically to explore, is my point.
Yeah, but it's a very Googleable. I mean, I've just Googled it and straight away, you know, between 0.74 and 4.5 million years ago is when they think this happened. And I'm sure we could do the same with the difference in the.
Well, I'd like to know why they think that.
Yeah. Okay.
Well, I'll say, I think this, I think that, but why? You know, well, I'm wondering if the Australopithecines, you know, how many chromosomes did they have? And the whole point I'm trying to make is, listen, we got some differences, some massive differences, let's say in the Y chromosomes. So rather than evolutionists just asserting like, well, maybe, you know, these deletions happened, or maybe this was duplicated, or maybe this was rearranged. Okay, anybody can say that. Anybody can make those assertions. I want to see that answered in more of an empirical fashion. And Catz, I mean this when I say it. Have the last word, and then pose your question to us. Maybe thermoluminescence dating would be a good one. I know Matt would enjoy engaging that one. But Catz, go ahead. Over to you, my good man.
Well, I would say if you want like an empirical answer, then I mean this took 20 seconds to Google. And this looks to me, you know, like it's got a fantastic explanation in it, scientific article. So the point I think I make is this information is out there. You know, it's not like it's not presented in science. It's not like it's not explained. It is out there. So, you know, where's the title? There we go. Have a look at that, and you'll find your answer.
And it's authored by Culture Catz.
Is it? Yeah, I've just done it now.
You're the man. You just did it. See, that's why we got you for this debate seminar, because you can write, get a paper peer reviewed and published all within 2 minutes. Catz, would you like to ask us? I think thermoluminescence dating, you brought it up a couple times. I think that'd be a good one. Isochron if you want, but Catz, the ball is in your court. You've been a good sport. What would you like to ask?
Well, I don't want to jump on thermoluminescence unless you're familiar with how it works, because if not, it wouldn't lead to a good line of questioning. Are you familiar with
your time? Feel free. I mean, I spent a couple minutes leading up to my question and you were patient, so feel free to have the same time, Catz.
You should. Right. Okay. Well, I'll explain it. Well, the main thing is, and before I explain, I'll just throw this out. When I was talking about examples like the Khufu ships, where we have thermoluminescence, which really has nothing at all to do with like isotope contamination or anything.
Actually, Catz, I just want to address one comment from Aaron. Appreciate it. I did not say, and if I did, it was a slip of the tongue. Nobody suggested that we evolved from chimps. What we're saying is humans and chimps share a common ancestor. That was neither a human nor a chimp. It would have been a common ancestor between those creatures. Anyways, Catz, I wanted to give you that opportunity to have a well-earned drink. So go ahead, continue.
Thank you. So when how well do you know? So, for example, Matt, how well do you know thermoluminescence dating? Do you know how it works? What the theory behind it is?
I know it's used for when they're testing forgeries, when they try to get a piece of pottery and they're like, it's been out of the sun, it's been out of radioactive waves from the sun, so therefore this amount of accumulated, so we should know how old it is.
Yeah, and actually, I've got a slide here about forgeries and how you can detect forgeries because they're very, very difficult to fake with artificial radiation. But anyway, the idea is that basically over time, when electrons get excited in a crystal lattice, they get trapped in little imperfections, and they can't de-excite back down to the normal state. So over time, and we can predict the rate this will happen over time, these trapped electrons will build up and up and up and up and up and up. So it's not, we're not saying that the crystal is radioactive or anything like that. We're just saying that these electrons get into an excited state. Now, when an electron de-excites, it emits a fluorescence, it emits energy. And what causes them to de-excitation to happen is when they're exposed to light. So if something is buried underground for a long period of time, it will build up charge. It's not charge, but let's imagine it's building up charge like a battery as an analogy. It builds up charge, builds up charge, builds up charge. Then it gets exposed to light and it fires that charge out. And it's not a current, but let's say we just measure it as a current, just as an analogy, right? We charge the battery, then we measure the current. And what happens there, when we have something buried underground, it will build up its charge, it will be exposed to sunlight, that charge will be lost, and so on and so on. Now, if we look at the charge, I keep using that term, but if we look at that, we can use the charge to figure out how long it's been since it was last exposed to sunlight, like you might do with a battery on your phone. I've got 50% charge on my phone. I can predict how long it's been since I've plugged it in, that kind of thing. So thermoluminescence will never age something older because it keeps getting reset. It will always age something under the age it's actually at. And when we use it for cave paintings, hundreds of thousands of years old and that kind of stuff. But anyway, that's the basis of it. So when we're using that to date pottery at the Great Pyramids, but it's being confirmed by dendrochronology, and then it's being confirmed by carbon-14. All these, dendrochronology, nothing to do with isotopes. These are all very, 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 very different techniques. So why are they all agreeing with each other from objects pulled from the same room? Would be my answer. It's a big ask, but...
Well, actually, I don't think we have too much of a problem with the pyramids. I might have said it earlier. I did. Basically, we actually like radiometric dating here quite a bit. I've got videos on it. We agree with the dates. Even we'll just say we completely even agree with the pyramids. The difference is we don't. Some people, like Answers in Genesis, they place the flood 4,360 years ago. Well, there's already a pyramid. If we're going to go by those dates. And that made me think, well, maybe that answers why the other version of the genealogies is more accurate, because there's an older version of these scriptures before the King James Bible, which came from the Masoretic text, called the Greek Septuagint, and it places the flood 5,365 years ago. Plenty of time for people to disperse and to create these things. So when I was looking at these radiometric dates, I saw that I was looking up to see if there was anything. Sorry, trying to share screen. It's totally frozen. It's okay.
Oh, do I need to pull my screen down first?
It's okay. Maybe that's what it needs to be. Sure.
Yeah, I'll pull that down. Sorry.
Okay. But you made a good point earlier. Are there some assumptions that go into this? Sure. I really wanted to know how accurate was it? They actually said the margin of error is 7 to 50%. That could take us back to the coin toss. But let's just go with those dates. We're completely fine with the age of that boat. There's nothing really that goes back man-made past 5,000 years. It's called the sapient paradox. And we notice that people just lived supposedly for 200 to 300,000 years without doing anything at all. And then in the last 5,000 years, they just did everything all at the same time. People, groups that never met one another, invented language out of nowhere. They invented writing systems, mathematics, all of it just appeared about it's exactly the same time. Very, very strange. Doesn't make much sense. And another thing that I really like about radiometric dating is the initial quantity, because before this, we had never really known what that was. We just assumed, like, I guess, just not much, probably no lead, right? Because uranium and thorium have to decay into it. And then we actually started to get into the Proton 21 laboratory experiments where they were showing the formation of these elements. And they were like, wow, look at that. Things are literally forming that we didn't expect them to. Like, look at the aluminum that formed, massive amounts of aluminum, but that comes through the decay process. Wait a minute, what's it doing there? So when we look at calcium 42, well, that comes through the decay rate of potassium 42, but this was made in a single day. So if that's made in a single day, that dated 104,000 years old. That's probably why we have a lead paradox, meaning like this core of the earth has way more radiogenic lead than there needs to be. So this paradox is literally solved by the observable evidence in the laboratory experiments that's showing these things literally forming right in front of our very eyes. And you notice that these laboratories like this one, they show that these radioactive elements lit up here are forming from these kind of like a cold fusion style of a thing. And they formed 2 radioactive ones right down here. So I wanted to look up, what are those? And sure enough, they're at the very bottom of a decay chain that normally takes a very long time to produce. So if we were to test that, it would immediately be old. So that's how I answer. Like, of course, they make great clocks, but we can't take our wristwatch and determine when it was made just by rewinding the watch. So, but the laboratory experiments answer why things are old, even though they're new and they're recent. So I know there was a lot there. I'm sorry.
No, sorry. Just to clarify then, with the boat thing that I was showing, and the thermoluminescence and the dendrochronology and the carbon dating, you're happy that they have combined together to correctly date the boat, because that could be after the flood, in your opinion. You're happy with that?
Yeah, I like that. That's how the longest living trees date the same. And it converges with that, right? I just
okay.
Yeah. Go ahead.
So sorry. Well, that leads me to the next question. And what it sounds to me like you're saying, can I just share my screen very quickly? So,
oh yeah, sorry. Let me take it down. It doesn't need to be there.
And sorry, this is why I said this at the beginning. It sounds, and even this, okay, good science always affirms scripture. If, in your opinion, the use of this dendrochronology and thermoluminescence and carbon dating in this instance doesn't, all combining together to give the same answer, if that doesn't, in your opinion, come before the flood, then it's good science, because it doesn't not affirm scripture, well, like actually, we had here. But if it was, we use these same techniques and it comes out to say, oh, this should have been before the flood, the earth is older, you're not going to agree with it. It sounds like, do you see my problem there? It's like, oh, I agree with it, which you've said. You've said that you agree with those 3 working together. You agree they work. But if I showed you any single example, even just one example that predated the flood, you would disagree with it.
Well, that's the thing. Dendrochronology, you really can't use it for something older than the flood, because the oldest tree is just 5,000 years old. So if you're saying, okay, I'm going to use dendrochronology and carbon dating to show that this structure here is 50,000 years old, well, that takes 2 things that we talked about earlier. If there was a flood, how would that affect carbon dating? And there's no tree to calibrate that with. So very true, but that you mentioned, yes, I agree with it until a certain range. Then I look for what would be the reasoning that this could be wrong. Is there a reason? Is there an answer? And if there is, like, for example, I did look at the Egyptian pyramid and I go, I think that this version of scripture is completely wrong. It doesn't match up. The trees are older. The living organisms are older. The population can't grow fast enough. And so I said, okay, it's not true. It doesn't match what I'm saying. So therefore, this version of scripture must be true. I use science to validate the version of scripture that would be true. Unlike a lot of people, they'll do the opposite. They'll say, no, I think the genealogies are wrong. I think there's reasons that trees have extra rings. I think there's reasons that, and they can have those arguments. I don't choose to do that. I actually choose to use the radiometric dates much more. So,
and I think that's where we differ. Like we differ and with all due respect, right? I did make this point in my presentation. I think that's where we differ, because you will agree that these techniques work. You will accept the results. But if the results say something come before the flood, dendrochronology or whatever, we can't use that anymore, even though it was correct another time, because I've already decided that the flood happened. So therefore, we cannot trust it, which is where, this slide came here. And I think that is the difference. That's what I tried to say early on. You will not accept it because you have the flood in your mind at a certain point. So therefore, if anything gives you a result before that, it has to be wrong, even if you trust the same stuff, to date stuff after the flood. And I think that's just where we kind of, part ways on the science.
No doubt. Go ahead, Donny. What are you going to say?
Well, I was going to say, because I want to respect Conspiracy Catz time.
Oh,
I cross-examined him for a little bit. Catz, you cross-examined Matt for a little bit. Did you want to give Matt the last word? And then we'll address just the super chats that came in real quick, power round, and then we'll call it a night.
Sounds good. It's been a great bit of fun. And over to you, Matt.
Matt, have the last word, and then I'm sure we could do this all day. Catz, you and I will schedule an informal discussion where we can kind of just dig deep, converse on ERVs and some of the topics brought up in the formal debate, and we'll be more relaxed informally. Matt, have the last word on that one, then we'll power through these super chats.
Okay. I just when you say I'll just agree with everything up until the flood. Well, the thing is I see the evidence being so parsimonious pretty much everywhere we look. Like there's a global flood narrative. There's Tower of Babel stories worldwide. When we go over here, we look at agriculture starting out in the Middle East. We see nothing in Africa, like they didn't do anything at all. And then all of a sudden humans create everything right there in the Fertile Crescent where the Bible talks about it. Language formations rise out of nowhere, tons of different language families. And then we see that there's writing systems, mathematics, all of it forming right there in that one region. I just see so much evidence with the mutation rates and the mutation accumulation in the mitochondria being so low instead of completely saturated. I see evidence everywhere. And then I go, well, that one piece over there says the rest of it's wrong. I'm like, I don't know. There's probably something like that is like, there's something else going on. I might not be able to explain it, but I'm going to get to the bottom of it. And that's what I try to do. So that's all I'm doing. Because it's I try to not ignore evidence. I literally make videos on every subject there is. So I don't want to be biased.
Gentlemen, we had some good feedback as well. People saying that they are going to want to listen to this again. And that's what we wanted. That's what we wanted for people. Catz and I, we've coordinated over probably a few months here to arrange this event. We wanted a comprehensive one for everybody, heavy on presentation and information and slides. Okay, gentlemen, we've got, looks like a couple questions for both sides. So we'll get through them. This one comes in from Eastgate Adoptionist. Appreciate the support. And it's for you, Catz. He says, paper released in 2022. Study challenges evolutionary theory that DNA mutations are random. University of California, Davis. The core of DNA is not random. Does this mean there is a designer for the original species animal? Any thoughts, Catz?
See, these questions where you say, oh, there's a paper just come out and it's not random. I can only give my gut feeling, and it's all it's going to be is a gut feeling, is that that's been written by a creationist, and it's going to be full of bias, and it won't be peer reviewed and accepted into a mainstream journal. Now, I'd have to read the paper, never seen it, but this idea that one person would say DNA mutations are not random, therefore there must be a designer, that to me doesn't sound scientific, but I'd have to read it. If you can get me the link, I'll read it.
Catz, do you think, based on this question, if a lot of mutations are non-random and perhaps environment drives certain changes and some mutations are more deterministic rather than probabilistic, in terms of some of the hierarchies that we see in mutations found in some of these genetic markers like pseudogenes, ERVs, if they're occurring non-randomly rather than randomly, is there any significance to, because then you could have two unrelated organisms non-randomly experience a mutation in the same location?
if things are completely non-random, I mean, my mind will go to epigenetics, which is obviously the effect of the environment upon gene expression. So you can affect gene expression, and that can, even epigenetics can be passed on to offspring. If I mean, I made a video a while ago about a population of starving people during the war, and that triggered certain epigenetical changes, which were then passed on via their offspring that weren't starving, etc. So I understand that the way genes can be expressed, which is maybe what this was on, can be environmental rather than random. I get that. If you're talking about, there's a base pair here and bang, just like that, across a species, it's going to be changed to this across the board, then obviously that's significant. And if I saw evidence that was happening, that will be a big problem.
Okay, I appreciate it. Matt, any thoughts on that one?
All I heard from him in the beginning was that this is a brand new study and he hasn't read it, so odds are I have not either. Kind of hard to
Matt, in your research, have you found that most mutations are probabilistic or deterministic or random, non-random? What are your overall thoughts on that as we engage the points that, hey, well, I should say this, as I said in my opening, we do have two very different positions, Catz, and we discussed this 4 or 5 years ago in our debate as well on genetics, that creationists, we explain the vast majority of DNA variation as being the result of created heterozygosity, as the evolutionists like to hear me say. And you guys would say that the vast majority is due to mutations. And so our job would be to determine what is a mutation, what is a designed allele. But we would both agree that mutations do happen. So for us, after the flood, mutations do take place. And if some fall out into hierarchical patterns, Matt, what are your thoughts on that?
Okay. I would say here, let me share screen real quick. I've got images for everything.
And then Conspiracy Catz, after Matt answers, this was for you. So you're going to get the last word. Okay.
Okie dokie.
Can you remove the super chat so people can see?
Yeah, go ahead.
Awesome. Thanks. So basically, here's an interesting thing. They found that when dogs mutated in the same regions as humans did, they got the exact same results in those sequences. So if the human lived in a city and they would get a particular type of cancer, the type of cancer the dog would get would be exactly the same thing. Very interesting. So that brings us to this one. The idea that mutations can occur randomly across genomes is widely accepted as true. However, recent research has revealed many factors such as nucleotide composition can influence the likelihood that mutations will occur at specific locations. Previous studies have also found evidence that DNA repair is targeted. So that similar things that we're seeing is more of like a design-oriented thing. It's not, you can kind of start to predict where these mutations are going to be. Completely opposite of what evolution predicted. It turns out that mutations are very non-random, and it's non-random in a way that benefits the plants. Within these mutations, a non-random pattern was revealed, counter to what was expected. Very, very interesting. So here we go. At first glance, we found what seems to contradict the established theory that initial mutations were entirely random and that only natural selection determines these mutations are observed in organisms. We also see it in bacteria. The beneficial mutations are very environmentally specific. Same thing with the cellular consciousness that they're talking about in this one. Scientists found a paradox in evolution. It might become the next rule of biology. And these things happen quick. Have you heard about the elephants that are being poached for their tusks? In one generation, they're being born without tusks anymore. How in the world does the elephant know, like, I'm being killed and then they're removing my tusks? How incredible is this, that they're now just being born without any? Kind of like humans. Our jaws shrunk because we're eating crazy food now where we shouldn't be, and our jaws are shrinking, so the body's not even producing wisdom teeth anymore. It's like it's smart. It knows what's going on. And then we know now that synonymous mutations that we once thought were silent are now increasing our knowledge that they're actually functional. And the type of function is beyond what they were talking about in the past. They didn't see that coming at all. Another great evidence for what we're talking about. New traits. This is a published study in 2010 that says most studies of recent evolution involve loss of traits, but we still understand the little about the genetic changes needed for the origins of novel traits. Over the broad sweep of evolutionary time, what we would expect to explain is the gain of complexity. Like, where is it in all of these organisms? All of them should be everywhere. We should find tons of evidence. And look, remember, 1.3% of mutations are beneficial. These are in plants. 75% are synonymous mutations, and the rest are harmful. And it's interesting, right? We're seeing, and so if the synonymous are also more harmful, what does that tell you, right? Just very interesting.
Matt,
yeah,
appreciate that. To be fair, Catz, we're going to now hand it to you. You get the last word because the question was for you.
I've got to read this one out from the chat. I mean, I'm not, but when you were talking about the elephant with the tusks, were you saying that they never developed tusks, or they were just born without them?
They started being kind of like humans. We stopped being born with them within just a couple generations.
Right. I honestly never thought elephants were born with tusks in the first place. I thought, like was said in the chat, I thought they grew them. But anyway, no,
I'm sorry. Like they grow them later in life, right? I guess what their development, kind of like wisdom teeth. We don't have the ability. They're being born without the bulbs to produce the wisdom teeth later in life, kind of like our wisdom teeth. That's all.
Got you. So yeah, sorry, I've kind of lost. Am I responding to that or just?
Oh, but you got the original question, I think.
Well, we can move on if you'd like, Catz, unless there was something you wanted to rebut based on what Matt said, or.
I can't remember what the question was.
I don't remember the original question either.
No, that's okay. Just for sake of time, we're trying to power through these. Catz, you've been so generous, as I know it's a lot later for you. This comes in from Clarence AS2. Thank you very much. This one's for us, Matt. If you like, you can give your thoughts. I can give my thoughts. $10 super chat. The Bible says a man can live 3 days inside a fish. Do an experiment on that, and then we'll see if the Bible is a reliable scientific source. My initial thoughts, and ICR documents this in an article, they say that there are fish large enough to swallow a man. So fact one, men have been known to survive inside a fish. Fact two, and the Bible says it happened. So based on the first two facts, that means what the Bible describes as happening doesn't necessarily have to be impossible. How that relates to our debate tonight? Well, our affirmative, Matt and I, was to provide convincing evidence for the Bible's claim that God created two people, Adam and Eve, just roughly 6,000 years ago. And at this point, we've gone for 4 hours. So it's up to the audience to watch the debate, consider the evidence, and see if Matt and I met our burden. Matt, how would you like to answer that?
I think Jesus himself referred to that as a miracle, because when he was alive, he said, since the time of Jonah, there will be no more miracles, basically until his time. So I think there was a gap. So God must have intervened to either keep him alive, or he died and was in the belly and resurrected on the third day. And that's why Jesus was pointing at him, because he, like Jonah, died, went to the belly of the earth, and then resurrected on the third day.
Okay. Short and sweet. Catz, I'm sure you don't agree with us on that one. So go ahead. What are your thoughts? I predict you're going to disagree.
my just viewpoint, my opinion would be this. Like, I don't understand, and it's kind of semi-adjacent to this question, but I don't understand why, given all the scientific signposts, things like that, etc., why a creationist won't say, okay, I believe God created the universe, right? But given the whole weight of scientific evidence pointing for evolution and old Earth, I believe he created it and life in this way. And I know this isn't directly answering the question, but it's made me think about this. I don't understand why the need to reject the scientific signposts, because it seems to me as if the God that you will believe in would have to be deceitful to leave those scientific signposts there. And that to me, I can never get my head around.
Okay, we'll get the final word since it was for us this time. My quick response would be, yeah, we're looking at the evidence, and we would argue otherwise, or else we wouldn't take this affirmative, right, Catz? That the scientific evidence actually suggests that what the Bible says about special creation is true. We've got evidence for one mitochondrial DNA ancestor, one Y chromosome ancestor on that mitochondrial DNA phylogenetic tree, which is just an unbiased look at real mitochondrial DNA variation. All mitochondrial DNA go back to 3 nodes. And maybe I could throw this at you if you don't mind, Catz. On that tree, I showed a couple times the lines connecting LM and N right in the middle. They're very short. Well, what lines represent on a phylogenetic tree or DNA differences or mutations. The lines radiating out, they're really long. So that would suggest, we would agree that that's a lot of mutations, right? But since there's only a couple separating the main nodes, why can't those be individuals? Why do they have to be, if I remember correctly, you said that they have to be populations. But again, they're only separated by a few mutations.
I would say there's no reason based purely on that and absolutely no other research whatsoever. If you're looking just at that representation that you've got there, there's no reason based just on that it couldn't be 3 people. I just think that the wider context says that it can't be.
Okay. And so if those could represent 3 people, and the Bible says that from these 3 couples, the earth was overspread, as a biblical creationist, wouldn't you agree that's kind of interesting, Catz? At least that point. If that point on its own, isn't that kind of interesting?
I can absolutely see why you are drawn to that. I can absolutely see why you're drawn to that because you've got the idea of the 3, I don't know the names, but the 3 women who come off the ark. And I can see why, from your point of view, you will be like, I'm going to look at that as evidence for my standpoint. I don't agree because I'm taking the wider context in, and there's, for me, even mitochondrial Eve wouldn't be one person. Sorry, it would be one person, but she wouldn't be the only person alive at the time. Just like these 3 nodes wouldn't be, even if they were individuals, not populations, they won't be the only 3 individuals alive at the time.
Appreciate that, Catz. Matt, any thoughts?
I do. I think that if evolutionists realize that since the very beginning in the formation of their basically the theory, that it literally just comes back down to horrible predictions to the point where like 99% have pretty much failed. This is a perfectly good example right here. What we're looking at, it's Ernest Mayr on the left. He predicted there would be no genetic similarity and distantly related ancestors. Here's the entire quote if you want to read it later. But he was basically saying there would be no homologous genes like a Hox gene for body layout plan and a PAX6 gene for your eyes. 1975 came along, the creationist model. He predicted the exact opposite would be true. And so when people go, well, genetic similarity, well, creationist prediction. Well, nested hierarchies, creationist prediction. And no biologist had even the foggiest notion that such similarities would exist in genes of such different animals. I could go down the list, but this idea goes back to Charles Lyell. He said, if we push this as far as we can go, we can prove to people that they're related to the orangutan. That was the original agenda. And so that's what they've been doing, is they've been retrofitting data after the fact. After the fact, they go, oh, well, what about hierarchies and homology? Oh, that was a creationist prediction as well. Oh, well, what about the founder effect? Creationist. What about the speciation rates? We predicted fast. Darwin predicted it would be slow. It's extremely fast. Anyway, I just am a huge fan of going, who predicted what first? The model should represent the better scenario for talking about the science.
Thank you, Matt. Okay, that one was for us. We'll move on. Here's the last one, Catz. It is for you. And again, Catz, I've really enjoyed tonight. I hope you've had as much fun as we have.
It's been a blast.
Eastgate Adoptionist, $10 super chat. Appreciate the support for tonight's debate seminar. And we were simulcasting, so I hope both channels enjoyed this exchange. He says DNA and RNA are ordered code. What did that code come from for the first living cell that can regulate and duplicate? Do ribosomes read RNA and create intricate proteins because they are sentient? What are your thoughts on that, Catz?
Well, I'll throw back to this because he's basically asking about, can I put this on screen? He's basically talking really about abiogenesis, isn't he? Can I share that? And like I said earlier on, abiogenesis, you've got a scientific point of view where we are looking at trying to replicate conditions of the early Earth to try and see how nucleotides might form with success on how these might form. Obviously, Miller-Urey is like the famous one, but a lot of people don't know how it's been built on with lots of others. we're not there in explaining exactly how this will have happened, but we are well within showing that. Oh, did I disappear for a second then?
No, you're good, Catz.
Oh, you did off my screen.
I did.
Sorry. So, we are well on track to showing that these really important molecules can form through natural conditions. So the question I put back at you is the counter argument. What is more scientific? Is it creating the conditions of the early Earth and showing that you can get proteins and amino acids and sugars, etc., forming literally, on their own, which we have shown? Or is it, something that can't even be tested, which is really the biblical model?
Catz, if you don't mind, I know you kind of responded to this in your rebuttal, but one of my biggest concerns are these mutually interdependent systems like DNA repair enzymes, because you're asking a good question. What's more likely? What's the most parsimonious explanation for the evidence? Well, something like your DNA repair system, where the information for DNA repair enzymes is in the DNA, and then you need DNA repair enzymes to repair the DNA for it to thrive and survive, and for us as biological organisms to propagate. And so where we would say the best explanation is special creation, because these both had to come into existence at the exact same time, how would you explain systems like that, these supposed chicken and egg systems? According to your model, what did come first, DNA or DNA repair enzymes?
Well, you'd never get to a big multicellular, long-living organism like us without a DNA repair system. So this is evolution. This feature has evolved very, very, very early on, hasn't it? With very, very simple single cell organisms with very short lifespans, and when these have evolved, then those lifespans have been able to increase. So it's not really a case of they're both needed at the same time. They are both needed if you want to have a big multicellular long living organism. But back when these things evolved,
are they both needed today in biological organisms?
They would be needed, for me and you and larger organisms, to have a healthy long life, absolutely. But if we're talking about single cell, the single cell organisms, you can't forget the ones that would have been at the early Earth are very different to the ones we've got today. If you want to have a single cell organism that is going to have the lifespan that they have today compared to what they would have had then. Yes.
So those ones that you're talking about in the early Earth, 1 billion or so years ago, is that where you'd put them?
Yeah, about that. But they would have had very fleeting short life, but it would have evolved, and it's natural selection. Those that evolved it would have lived longer and passed those, past those traits on.
Isn't that the story rather? So would you say that those pre-cells or primitive cells, they evolved, therefore they're no longer here? Like, you couldn't point to one of those today and say, hey, see, here's an example. Here's a single-celled organism that reflects that primitive organism from 1 billion years ago? Or do you kind of just have to appeal to the unobserved distant past?
I don't know. You've got to remember the conditions of the early Earth were very, very different.
I wasn't there. I don't know. I don't have a memory of them.
Okay, so the conditions of the early Earth, the temperature, the atmosphere, all of those things were vastly different than today. And if you want to think of it as some kind of extinction event that would have got rid of, plenty of historic life, because they just, weren't evolving fast enough. I suppose it did evolve fast enough because we're here, but I wouldn't expect organisms that were suited to an environment that's extremely different to still be here in an environment that is completely different.
Well, that's my point. If today organisms require these mutually interdependent systems, and so we as creationists say that means they had to have been created that way because they need those systems at the start, at the beginning to function. But you're saying, well, 1 billion years ago, there were primitive organisms that didn't have those systems. But can you show us a fossil of that organism and show us the genetics and say, hey, see, here's that primitive organism I'm talking about? Or is this just more of an appeal?
Well, single cell organisms aren't going to form fossils.
Okay, no, I understand.
What I can do is, again, just repeat what I said before. We can look at, is it possible? Can we mimic the conditions and see the formations of the things that would be required to put the cell together? And we can. But can we, in any way, shape, or form, see a talking serpent or dust somehow just immediately? And if that's what people believe, that's what people believe. But it's not scientific. It's not testable.
Whereas, isn't this something you believe, that 1 billion plus years ago there were primitive cells? See, once you believe the first sentence of the Bible, in the beginning God created, then miracles are possible. They're not hard to believe. But yeah, if you have a naturalistic worldview or starting point, then the miracles that you read in the Bible, those are things you're not going to be able to believe, because your worldview and your naturalistic starting point prevents you from believing those, right?
I get where you're coming from with that, because I know you will have a very strong belief in the Bible. So for you, it's almost taken care of. You know what I mean? Like it's all taken care of. If there is a problem with the thermodynamics because of the flood, or if there is a problem with abiogenesis or whatever, well, it's taken care of because you've got that faith, you've got that belief. The science has to figure it out. But what we can do is we can do what we're doing. We can look at genetic analysis. We can look at fossil records, and we can start to piece back. And as soon as that tree falls apart, as soon as we start piecing all this stuff back in it and it doesn't work and things don't start to gel, then we've got a question. But actually things are gelling together and they are corroborating each other, which is, I would rather believe evidence that corroborates rather than the other way around.
And of course, based on the last 4 hours, we would say that the evidence corroborates our position. You say the evidence corroborates yours. And I guess at this point, we leave it up to the audience. But Catz, I do appreciate you interacting with those questions. This has been good. So we'll give you the last word there, because that question was for you. And I almost missed this one. Sophia, I think this relates to your guys' cross-exam on dating methods. So
it was what I was saying. I showed the chart where they had produced calcium-42 in a laboratory, but it's of course, yes, it is stable. That's the whole point, is potassium-42 decays into calcium-42, and that's the decay chain. So how in the world is it being produced in a single day in a laboratory when it's supposed to take hundreds of thousands of years to form through a decay chain process? So if it's being formed and we dated it a day later, it was dating hundreds of thousands of years old and it was formed a day before. That's all I was saying. Yes, it is at the bottom of the decay chain. It's like finding a bunch of lead in a zircon crystal and going, wow, how'd that get there? It must have came from uranium and thorium only. Rather, wait a minute, what if the whole decay chain formed and in higher proportions of the smaller decay chains, and they're rapidly decaying, making all that lead? That's what we saw in the laboratory. So that's what makes it so fascinating. Anyway.
I've got a real quick question on this, just to go out with a bang. For Catz, you always have informed answers, and so I appreciate asking you these technical questions. What are your thoughts on, in the world of dating methods? Because I like to say all it takes is one that shows us a young Earth to overturn all the ones that are interpreted to point to an older Earth. And one that I like to point to, and I'm curious as to your thoughts, if you're familiar with it, is the helium that we find in these zircon crystals. And so in a nutshell, these zircon crystals are supposed to go back 1 billion or so years, and uranium to lead releases helium atoms, but helium is a very slippery molecule. And we've looked at the rate as to which helium dissipates. And if those zircon crystals were 1 billion or so years, we shouldn't find the amount of helium that we do find in them. We find quite a bit. Dr. Russell Humphreys did a study on it, because helium it leaves so quickly during the uranium to lead decay process that there shouldn't really be all that much there. But we find a lot there, suggesting that the leakage hasn't been taking place that long. Catz, any thoughts on that?
Well, I mean, how many steps, to think back to my A-level physics, how many steps in that decay chain are producing alpha particles? Can you remember how many steps
I think
between
8.
uranium and how many? Sorry,
I think 8.
When you're saying it should just like leave instantly.
Not instantly, but we do know the diffusion rate, and it's quite fast. Dr. Russell Humphrey's paper, he looked at the known diffusion rate. I could pull it up. And he got an age of somewhere between 6,000 to 10,000 years on known helium diffusion.
Yeah, I mean, I'd have to have a look at that. I'm not quite familiar with the argument that you're making to give an answer on that, but basically you're saying that you've got these minerals and you believe that the amount of alpha particles that are given off in there should have dissipated much more quickly than they have when it comes to aging.
Yeah, like right here, you got 8 helium atoms, right? You're going through the decay process, uranium to lead, but helium is so slippery that we shouldn't find as much helium as we do find. So the argument is there's too much helium in these rocks for them to be millions and billions of years old. Here's the paper: helium diffusion rates support accelerated nuclear decay. And right here, he says, this is not in the paper itself. That's very technical. This is kind of just a layperson rundown of it. And he says, though the rocks contain 1.5 billion years worth of nuclear decay products, newly measured rates of helium loss from zircon show that the helium has been leaking for only 6 plus or minus 2,000 years, because there's just too much helium there still. Because if that was leaking out for a billion plus years, then of course you're always releasing a little bit of helium. So there should be, I guess, a little bit, but not as much as we do find. And of course, if you're not,
I mean, the elephant,
that one, that's completely fine. But here's the paper if you wanted to look at it later.
The elephant in the room there, though, is he is saying that they contain products from a billion years of decay.
Right.
He's saying that.
Yeah, that's the interesting point. So his conclusion, and Matt, I believe you have a different conclusion, but his conclusion is, okay, so we got 1.5 billion years worth of nuclear decay from uranium to lead in these rocks. But wait a minute, we've got the amount of helium consistent with 6,000 to 10,000 years. So how do we answer that? Well, his solution is, okay, that means at some point in the past, either during creation or during the flood or both, you would have had accelerated rates of decay, which explains all of the nuclear decay products in a short amount of time in order to retain the helium that we find in those rocks. Does that make sense?
Yeah, I'd be interested to see how he's come to that conclusion, because obviously the decay chain doesn't work, that all the uranium, something decays to become the next element, and then that all decays to become the next. It's a random process, and you will still have some uranium left when other atoms are like this far down the decay chain. And I'd be interested to see how he's come to that prediction. I really would be, because we might know how fast helium diffuses, but we've got to remember that at some point you're going to have all those elements present. And if you said there's 8 of them that are giving off helium, how long are they going to be giving off helium for? I'd be interested to have a look at that. If you can send us a link to that, I will be interested to have a look.
Absolutely. And then in a follow-up informal discussion, we can touch on that. And I think a podcast is much needed. You and I, Catz, going down deep, staying down long, coming up dry on ERVs. We can do a couple hours of that. So, okay, gentlemen, I don't know about you. I'm pretty tired. 4.5 hours. You guys look like you're ready to go for another 4.5 hours. I really appreciate this. I had a ton of fun, and I think we got a lot of information out there. I'm glad we got a good hour of discussion and audience question and answer time as well. Catz, thank you for doing this. Any final words, final thoughts?
No, it's been great. It's been a lot of fun. And yeah, let's do it again.
And Catz, you get a good sleep, because I know it's really late for you. And
it's 2:45, yeah.
Have to work in the morning. So Matt, thank you for doing this. Matt, you're kind of dark right now. You're like shadow man. So it's probably getting late for you. Final words from you, Matt?
No, have a good night, Catz. Sleep good, man.
Night. Thanks.
Thank you so much. To the audience, we'll chat soon.